those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started.
All right, welcome to Standing for Truth, everybody. I am your host, Donnie, and today we have an extremely exciting program that will focus on junk DNA. We are going to be answering many very, very important questions about junk DNA and how exactly it relates to creation and evolution. Looks like we've already got a great chat. Thanks, everybody, for showing up. And Dr. FuzRana, thank you so much for giving us your time for this important presentation. I really appreciate it.
Well, Donnie, thanks for having me. It's always a lot of fun to talk about these kinds of issues.
Amen. Amen. Well said. It's certainly a privilege, Dr. Rana. So a few of the questions we're going to be addressing today is junk DNA still a valid argument in favor of evolution? Does most of the genomes of living organisms, including humans, consist of evolutionary leftovers, genomic fossils, and genetic baggage? Why would God create genomes of junk? Wouldn't a designer create genomes of treasure and function? These questions and more will be answered today. Now, before we get right into your presentation, Dr. Rana, since it's your first time on the show, I do want to give the audience a brief introduction into who you are and how they can find more about you, because I've spent hours now reading through your articles and I highly recommend them. So biochemist Fuz Rana is VP of Research and Apologetics at Reasons to Believe, an organization dedicated to communicating the powerful scientific case for God's existence and the Bible's reliability to Christians and non-Christians alike. Fuz earned a BS in chemistry with highest honors, West Virginia State College, and a PhD in chemistry with an emphasis in biochemistry from Ohio University. He pursued postdoctoral studies on cell membranes at the universities of Virginia and Georgia, and worked as a senior scientist in research and development at Procter & Gamble. Fuz has been published in peer-reviewed scientific journals such as Biochemistry, Applied Spectroscopy, FEBS Letters, Journal of Biological Methods, and Journal of Chemical Education. He also holds 2 patents, authored a chapter on molecular convergence and intelligent design for the nature of nature, and co-wrote a chapter on antimicrobial peptides for biological and synthetic membranes. In addition, he has delivered numerous presentations at international scientific meetings. In other words, Dr. Rana, you're more than qualified to be discussing these topics. I appreciate it. Is there anything else you kind of wanted to add there? Anything that I may have missed, Dr. Rana?
No, that was a very generous introduction, so nothing to add.
Okay, awesome. I appreciate it. Check the description box, everybody, for more in terms of Dr. Rana's work and articles he's putting out. So that being said, I'm going to hand it over to you, Dr. Rana, to get right into the topic and the presentation for this evening.
Yeah, and I'll just do, I'm hoping about 30 minutes or so of a presentation, and it's not going to be complete by any means, but hopefully we'll fill in the gaps during the discussion in the Q&A. And I'm going to go ahead and share my screen to get us started. And I'm not quite sure how to do this any more eloquently than I am right now. So here I am bumbling around, but all righty, here we go.
We got it. Looks good.
Okay, thank you. Yeah, and what I thought I would do in terms of addressing the issue related to, quote unquote, junk DNA is to take a look at the ENCODE project, because this is a body of work that I think has really important bearing on the question of the structure and the function of not only the human genome, but really by extension, the genomes of other organisms. And, I don't know if anybody remembers, I think it was June of 2001, or no, June of 2000, if I'm not mistaken. My, how the time flies. When Bill Clinton, then President Bill Clinton, hosted a press conference where you have Francis Collins, who headed up the Human Genome Project, sponsored by NIH, and Craig Venter, who was the head of Celera Genomics, a private company competing with the public project to essentially sequence the human genome, where they realized that whoever got across the finish line first had a lot to gain by that accomplishment, where Venter, through Celera Genomics, is really trying to commercialize the information that was found in the human genome. And they ended up agreeing to lock arms and to call a truce to their competition, share information with each other, and collaboratively completed the first draft of the human genome sequence. And at that time, then President Bill Clinton had these remarks where he said, "Today we're learning the language in which God created life. We are gaining ever more awe for the complexity and the beauty and the wonder of God's most divine and sacred gift. With this profound new knowledge, humankind is on the verge of gaining immense new power to heal. Genomic science will have a real impact on all of our lives, and even more on the lives of our children. It will revolutionize the diagnosis, prevention, and treatment of most, if not all, human diseases." And of course, the human genome is about 3.2 billion genetic letters in size. It's a massive amount of information. And this is a picture of me a few years ago at the Wellcome Trust Museum in London, England. And in the Wellcome Trust Museum, they have a display of the human genome. And they have these various volumes that essentially are listing out the human genome sequences. And the volumes are numbered according to the chromosome number for each of the sequences. And you can see how much information is harbored in the human genome when you look at 3.2 billion genetic letters. And of course, you know, we have all of this information, but the question then becomes, what does that information actually mean? All right, how do we really make sense of this information? And along those lines, some of the very first work was to try to identify the number of genes in the human genome. And to everybody's surprise, there's only about 20,000 genes in the human genome. There are techniques you can use where you can kind of identify certain sequence markers that indicate the beginning of a gene sequence. And using that technique, estimates are there's about 20,000 genes in the human genome. And this is a far smaller number than anybody would have anticipated, which indicates that a vast proportion of the human genome is consisting of sequences that are non-coding, that don't directly code for protein products or for RNA products like ribosomal RNA and transfer RNAs and things like that. And so this insight from the human genome sequence at that time fit perfectly within the confines of evolutionary biology, where people argued that most likely 90 to 95% of the human genome consists of non-functional or, in colloquial terms, junk DNA, and that this finds explanation in something known as the C-value paradox. And the C-value is a parameter that refers to the amount of DNA in the nucleus of an organism's gametes, measured in picograms. And you could think of the C-value as roughly equivalent to the size of the genome. And it turns out that when people began measuring the C-value, they expected initially to see some kind of correlation between genome size and the complexity of organisms. And in fact, they saw nothing of the sort, but rather there didn't appear to be any kind of relationship whatsoever between C-value and, again, organismal complexity, where some relatively simple organisms had very large genomes compared to much more complex organisms. And The ultimate resolution of this was to argue that most of the genomes of organisms are actually consisting of non-functional or junk DNA that was accrued in the genomes of organisms through a variety of different mechanisms, and that these junk DNA sequences actually are highly informative because they can give us a picture of the evolutionary history of organisms, that these are vestiges of an evolutionary history, and that the relationship or the lack of relationship, again, between C-value and complexity is really a reflection of the junk DNA content. And when it comes to the human genome, again, it looks like somewhere between 90 to 95% of the human genome consists of what appear to be non-functional DNA sequences, where a significant portion of the genome consists of what are known as retrotransposons, line and sign and long terminal repeat sequence elements. There's a very small portion of the genome that consists of exons, which would be the coding region. A significant portion are introns, and then there's some satellite DNA sequences, particularly associated with centromeres and with telomeres. But again, it looks like, again, a significant portion of the human genome is indeed non-functional. And in fact, we've identified the different classes of these so-called non-functional sequences. And with each class, we've identified what we believe to be mechanisms that could account for the origin of these different types of sequence elements into the genomes of organisms. And so for many people, junk DNA really represents one of the most compelling reasons to think that human beings must have an evolutionary history, and that evolutionary history is shared with the great apes and in other organisms. Because again, people are arguing that these sequences are the vestiges of evolutionary history. And of course, this begs the question, why would a creator create genomes of organisms where a vast proportion of the genome is non-functional? But more critically, why would a creator create genomes of organisms where not only are there a large proportion of non-functional sequences, but those non-functional sequences are shared with other organisms that would naturally group with, in this case, with human beings, like the great apes? Why would these sequences have such a high degree of similarity? Why would these sequences occur in corresponding regions in the genomes? Why do these sequences display a nested hierarchy where you could actually organize these sequences into groups of groups that presumably reflect an evolutionary history? And then also, why would these sequences, in the case of endogenous retroviruses or the retrotransposons, why would they bear similarity to viral sequences, viral genetic information? Or in the case of pseudogenes, why would these pseudogenes display similarity to quote unquote functional or healthy genes? And so these are really questions that are profoundly challenging to intelligent design, to some kind of creation perspective. And it makes sense, particularly if you are embracing a materialistic view of origins, to look at the patterns that we see with junk DNA as being compelling evidence for common descent. And so if we're going to respond to the junk DNA challenge and present a creation model, if you will, or a design model for genomes, we've got to address all of these challenges. We have to be able to show that these sequences actually are functional, that there's a reason why these sequences would be shared, that there's a reason why they would be organized as nested hierarchies, and that they bear similarity to, again, things like to retroviral sequences or to healthy genes. So this is really the task that's in front of us if we are going to make a case that the human genome reflects the handiwork of a creator. And there's a lot of different ways to unpack this challenge and to marshal a response to the junk DNA challenge to the design argument. But what I'm going to do in the presentation is really focus on the functionality of DNA and of non-coding so-called junk DNA. And what I'll then do is bring in some comments about these other factors as we go along the way, but we can leave some of the details to our time of discussion and time of Q&A. So I understand that the presentation I'm going to provide here is not complete. It's only really a partial response. Now, the ENCODE project, you could think of this as being kind of like a quest for the Rosetta Stone for the human genome. And this project was launched shortly after the completion of the human genome sequence, or at least the first draft, where the goal of the ENCODE project was to try to catalog all the different functional elements in the human genome. And so ENCODE stands for the Encyclopedia of DNA Elements. And this project began as a phase 1 project, which was a pilot level study that looked at a small proportion of the human genome. And the initial results were rather surprising because it looked as if a significant portion of a small percentage of the human genome actually consisted of sequences that were serving some kind of functional role. And the assays that were used in phase 1 included measurement of transcription, with the idea that if sequences are being transcribed, they most likely are functional. The binding of transcription factors, the binding of histone, the modified histone binding. This is related to epigenetics. Methylation, again, related to epigenetics, where again, the idea is that these interactions are serving a gene regulatory role. And then also assays that looked at three-dimensional interactions with respect to components in the human genome. And in, I think it was 2011, phase 2 of the project was published, where a large number of different cell types had their genomes analyzed for functional DNA. There's probably about 200, 210 different cell types in the human body. I think in phase 2, somewhere around 140 to 150 of these cell types were evaluated. And what was shocking to everybody is it looked as if 80% of the human genome displayed some type of biochemical function that was interpreted as having actual function, that a significant portion of the human genome was functional. And at that time, Ewan Birney, who was heading up the ENCODE project, was interviewed by Ed Young for Discover, and he said, what's in the remaining 20%, possibly not junk either. It's likely that the 80% will go to 100%. And we now have seen phase 3 and phase 4 completed. And while I don't know what the percent is for functionality, it really does look like, as ENCODE project workers continue to fill in the missing gaps, looking at functionality in other cell types, as well as functionality at different times during the course of development and during the course of the cell cycle, that at some point, most of the DNA in the human genome appears to play some kind of functional role. The phase 2 project indicated that there were 4 million regulatory regions in the human genome, and these included 70,000 promoter regions and 400,000 enhancer regions. So the number of regulatory elements in the human genome was far greater, and the implication here is that many of these regulatory elements are actually residing in portions of the genome thought to be junk, like long terminal repeats or the line and the sign sequences. In addition to the ENCODE project, there have been other studies that have been done that have discovered in a piecemeal manner functionality for different types and different classes of junk DNA. So for example, about 2% of the human genome consists of pseudogenes, which were believed to be the remnants of once functional genes. And there's different types of pseudogenes, unitary pseudogenes, duplicated pseudogenes, and then, of course, processed pseudogenes. And it turns out that these pseudogenes actually seem to be playing a critical role in gene regulation through what has been dubbed a decoy mechanism. In fact, recently there's been an idea that's been advanced called the competitive endogenous RNA hypothesis, which actually provides a really elegant framework for understanding not only the function of pseudogenes, but why pseudogenes bear similarity to their quote-unquote functional, or maybe their coding counterparts. And it has to do with how the decoy mechanism works. But this competitive endogenous RNA hypothesis explains functionality for not only duplicated pseudogenes, but unitary pseudogenes as well. And we're also discovering that some pseudogenes actually code for functional proteins. Now, one of the criticisms that is leveled against arguing that pseudogenes are functional is that, well, some of the pseudogenes are transcribed, and therefore, you could argue that they are functional, but a number of pseudogenes actually are not transcribed. And it turns out that there may actually be functional roles that these untranscribed pseudogenes are playing as well. But part of the challenge here is methodological, is that many times people are assuming regions or assuming pseudogene sequences are not being transcribed when in fact they are. And the challenge is it's very difficult to distinguish between a pseudogene and a coding gene sequence through some of the assays that are used to measure transcription. Line DNAs have been observed to play a role in X chromosome inactivation, as well as play a role in what's called monoallelic gene expression. Sign DNA is expressed during the cell's stress response, and it looks as if these sequences are playing a protective role as well as a role in gene regulation. Endogenous retroviruses look as if they actually play an antiviral role, and the way in which they exert their antiviral function is actually multivariate, that there's a number of different mechanisms, but all of these mechanisms actually are predicated on the similarity between these endogenous retroviral sequences and the genetic sequences of both retroviruses as well as other types of viruses. Elements of endogenous retroviruses, like the long terminal repeats, also play a role in gene regulation. Just recently, a research team indicated that these endogenous retroviral sequences actually play an anti-cancer role. That P53, the guardian of the genome, actually, when cells are stressed, which would take place when cells become tumorigenic, the P53 actually triggers the expression of endogenous retroviruses, which triggers essentially a response on the part of the immune system to clear those tumor cells from our bodies if we have cancer. So the bottom line here is that we're discovering that junk DNA is anything but junk, that it's actually functional DNA. And in terms of these shared sequences that we see, you could actually argue that the shared sequences are reflecting common design as opposed to common descent. And that we could also explain nested hierarchies through a common design model, if you will, a shared design model. And we'll get into the details of that if people are curious. I'm going to go ahead and skip ahead here and just close out the presentation by saying that I think it's legitimate to say that there's a new view of the genome that is emerging, that we really are in our infancy when it comes to understanding the human genome as well as the genomes of other organisms, and that it's very easy in the absence of having this understanding to draw conclusions about the genome that seem to indicate that most of the genome is non-functional. But as we're continuing to learn more about the genome, we realize that much of the non-coding regions of the genome that were dubbed to be non-functional or junk actually are serving very important roles in terms of regulating the expression of the coding sequences. Eric Green, who was the director of the National Human Genome Research Institute, when phase 2 of the ENCODE project was published, said, during the early debates about the human genome project, researchers had predicted that only a few percent of the human genome sequence encoded proteins, the workhorses of the cell, the rest was junk. We now know this conclusion was wrong. ENCODE has revealed that most of the human genome is involved in the complex molecular choreography required for converting genetic information into living cells and tissues. Okay, so I'm going to go ahead and stop there, and that hopefully just sets the stage. We can go ahead and just open it up for discussion and for a time of Q&A.
Awesome. Dr. Rana, that was a fantastic presentation, and so much good information. I know it's hard to just condense into a 30-minute presentation. That's why I want people to definitely check out the links in the description box for more on this and more on this in incredible detail. And Dr. Rana, you asked and answered all the right questions. Why would pseudogenes display similarity to functional or healthy genes? And why do we see nested hierarchical patterns in terms of these pseudogenes, junk DNA, and endogenous retroviruses? Is this a reflection of descent or design? I think you did a great job demonstrating why this can certainly demonstrate design due to the function that we're finding. So my first question, we got a ton of questions from the audience as well, Dr. Rana, but my first question after watching your presentation has to do with the ENCODE results. And we know that there's been a lot of evolutionists who have kind of been anti-ENCODE and have presented some challenges. What are your thoughts on that? And then how do we best address those challenges?
Yeah, and it's interesting because shortly after the ENCODE project published the phase 2 results, it initiated a significant backlash on the part of many evolutionary biologists who really decried the results of phase 2, where they argued that this would undermine the C-value paradox, that key tenets of evolutionary theory would be undermined if this indeed was correct. And the argument was that the ENCODE project scientists didn't really understand evolutionary biology to make these kind of outlandish statements. And so it ignited what you might call the junk DNA wars. And I think Carl Zimmer a few years ago wrote a really nice article, I believe it was for the New York Times, where he described the whole junk DNA wars, the junk DNA wars that was happening, not between ID proponents and evolutionary biologists, but within within the scientific community itself, where scientists who work in genomics found the results of the ENCODE project to be very helpful, whereas the evolutionary biologists again decried, the ENCODE results. And I a number of years ago did a debate at North Dakota State University on Darwin Day, where I engaged P.Z. Myers, and I think some of you may be familiar with P.Z. Myers. He has an award-winning science blog called Pharengula, and is an outspoken atheist as well. He's at the University of Minnesota Morris. And during our discussion time, a question about the ENCODE project came up, and what P.Z. Myers said was that the results of the ENCODE project most definitely can't be valid, because if they were, then it means evolution or key aspects of evolutionary theory simply can't be correct. And so it was interesting to me, because at that point I realized that what was happening was that the data was being evaluated in light of the theory, as opposed to a theory being evaluated in light of the data. And so it was almost like a bizarro world approach to science. And that attitude I see among many of the chief critics, of the ENCODE project results is they really see the results as being flawed because of the implications for the evolutionary framework. But there's been a number of specific challenges, and we can kind of get into some of those if you would like. But that essentially kind of sets the tone, I think, for what you're looking at with respect to the junk DNA wars is really two very fundamentally different views of the human genome that are at odds with each other, even within the scientific community.
Right. That's a great, those are some great points. And Dr. Rana, in terms of challenges, because, and correct me if I'm wrong, what we discovered, or one of the things we discovered with the ENCODE project is that most of the genome is transcribed in RNA. I think it's about 80%. And the question becomes, if 80% of the DNA is transcribed, is this activity useless activity or beneficial? And I've heard a lot of evolutionists say, well, a lot of this activity is just spurious, or it's not really doing anything. It's just easier for the cell to just transcribe it because it would take more work to not transcribe it in terms of the cell. And so they'll say that this low-level transcription can actually be beneficial for the cell, even though it's not really performing anything beneficial or functional. What are your thoughts on? Have you heard that response before?
Yeah, and that's a specific example of maybe one of the most, I think, important critiques of the ENCODE project, and it's a critique that I think needs to be taken seriously, and that is, if we're measuring biochemical activity, does that activity actually translate into function? And I've seen this primarily played out with respect to transcription, as you're pointing out, as well as to the binding of transcription factors to the genome, where people are arguing that much of the binding is just simply random binding at non-functional sites. And to me, as a biochemist, when I hear people argue that a vast proportion of the transcription that takes place is essentially spurious, as you said, or just biochemical noise, that to me doesn't sit very well, because to transcribe a portion of the genome is a very energy intensive process. You basically, every time you're adding a nucleotide to a growing RNA chain, you're consuming 14 kilocalories per mole of energy. That's a lot of energy that's being released as heat for no purpose whatsoever, just to create transcripts. Not to mention that these transcripts would essentially clutter up the interior of the cell if they're not playing a functional role. So right away, that doesn't intuitively make sense to me. But as people have actually cataloged the different types of transcripts that are produced, it's very easy to show that these transcripts are actually playing a functional role. And so, for example, 2% of the transcripts are for protein, are protein coding. 2% are pseudogenes, which are playing a regulatory role. But there's a number of transcripts that are being transcribed, even though they're not translated, but they're playing a regulatory role. And these would include things like long non-coding RNA molecules and in small interfering RNA molecules and MicroRNAs and things like that. And we know that these are again all playing regulatory roles as people pursue, the functionality of these transcripts. It appears as if they're playing regulatory roles. So it doesn't look like, with regard to transcription, that this is actually, a valid critique.
That's a great response, Dr. Rana. To me, it would be, as you pointed out, it would be a waste of energy and resources for the cell to transcribe all that information, which really just turns out to be spurious and useless. And the question that comes to my mind is, if this really were the case, wouldn't natural selection over millions of years of evolutionary history have removed all of this junk or all of this information if it really was just spurious?
Yeah, well, because that's a very good point, Donnie, because there are mechanisms that will suppress transcription. And so you would think that if you were really transcribing that proportion of the human genome or the genome of another organism, and it's biochemical noise, that there would be, evolutionary processes in play that would essentially suppress that transcription, that we would expect transcripts that are playing no role at all to be rather rare, not to be occurring at that high of a level.
Right, great point. And you mentioned P.Z. Myers, and I've read in a book, I believe it was by Dr. Lee Spetner, but he pointed out that Richard Dawkins, and I've actually got the quotes in front of me. In 2009, he said, "It is a remarkable fact that the greater part, 95% in the case of humans, of the genome might as well not even be there for all the difference it makes. But after the ENCODE result and a lot of the results have come about that you've been pointing to," he then stated, years later, I believe in a debate with Jonathan Sachs, he said, "I know there are some creationists who have jumped on it because they think it is awkward for Darwinism. Quite the contrary, of course. It is exactly what a Darwinist would hope for, to find usefulness in a living world." So to me, it kind of seems like they want the best of both worlds. If it's junk, it's evidence for evolution. If it's not, it's what they hope for, judging by Dr. Dawkins' quote.
Yeah, it's interesting because I've been at this for a while now, and I can remember even before I started working at Reasons to Believe, which was in 1999, that the junk DNA challenge was present and was raised against the design argument, and I didn't see anybody at that time making any kind of predictions from an evolutionary perspective that junk DNA was functional. In fact, it was the opposite. And very few people actually even bothered spending time studying, junk DNA sequences. A few people might have cataloged them and maybe did some work in terms of trying to understand how these sequences may have originated, or they may have used them as tools for phylogenetic analysis, but nobody really ever even thought to probe them for functionality. In fact, Craig Venter, who we saw a picture of earlier, was at one time part of the Human Genome Project. And he actually argued that it was a misguided approach to sequence the human genome because most of it's non-functional. What we ought to be doing is developing assays that measure transcription in the human genome and then characterize the transcripts, because only those sequences that are transcribed are going to be functional, and that we ought to build up our knowledge of functionality based on transcription. And in fact, published a landmark paper where he looked at transcription in neural tissue and discovered a huge number of unknown genes through that process. And was so frustrated by the approach taken by the Human Genome Project that he actually left and began pursuing his own research efforts along those lines in the private sector. But all that to say is that, people, when the Human Genome Project was underway and when the first draft was completed, nobody was making predictions about function. And in fact, if you look at anybody that was making predictions about function, it would have been people that held to an ID or to a creation, model approach. In 2000, I wrote an article where I put in print that we would expect the different classes of so-called junk DNA to one day turn out to be functional. And so it's only later on, when function is being discovered, that I'm now seeing, evolutionary biologists say, well, evolutionary theory predicted this all the time. And I think it's okay to say, look, there's, neo-functionalization, I think is not unreasonable, where the argument would be, hey, all of this so-called non-functional DNA is raw material that evolution can co-opt and modify and make use of to create new functionality. Okay, that's not an unreasonable argument. And and neo-functionalization it seems to be a reasonable explanation when you're talking about isolated examples of functionality. But when you're talking about this wide scale level of functionality, that's where I think neo-functionalization is really problematic. It's one thing to say, hey, a handful of pseudogene sequences turn out to play some kind of regulatory role, and that this could be explained by neo-functionalization. But to say that the totality of pseudogenes is actually playing an integral role in gene regulation, to me, really makes it hard to argue for neo-functionalization at that point, just because of the nature of the evolutionary mechanism, which is unguided and undirected and historically contingent. So if neo-functionalization is your explanation, that works well when a fraction of the sequences are functional, not when the totality or the near totality of those sequences is functional.
Well, so many great points there, Dr. Ron, and I'm glad that you addressed the argument pertaining to neo-functionalization. And going back to a few things you said, which I completely agree with when it comes to predictions, testable predictions. As you've pointed out, it's the intelligent design advocates that can look to a lot of these results, for example, from the ENCODE project, suggesting so much genome activity. Intelligent design advocates like yourself can make future predictions on function pertaining to pseudogenes or retroviral-like elements, including ALU sequences. But from my understanding, it's the evolutionary community that are more so silent in terms of making predictions on function.
Yes, yeah, and again, it's only recently where you begin to see, people saying all along we thought this was functional, right? And and of course, I mean, again, to be completely fair, everybody thought that a portion of the non-coding DNA was functional. Right. These would be the regulatory elements and things like that. So, everybody agreed that there was going to be a portion that was non-functional. But I think the shock is the level of regulation that the non-coding DNA or the so-called junk DNA is actually playing or contributing. That to me is I think what is shocking. And in terms of predictions, in 2004, check that, 2005, when we published the first edition of Who Was Adam, we addressed the junk DNA issue. And again, there was very little that we could really say at that time, about junk DNA. But along the lines of endogenous retroviruses, I went back just recently and was rereading that section of the book, and I actually made a prediction there about endogenous retroviruses not only being functional, but actually playing an antiviral role, serving as part of the innate immune system. And it turns out that in recent years, there's just a flurry of papers that are now being published showing that endogenous retroviruses, as well as endogenous viral elements, EVEs and ERVs, are playing a very important role as part of the innate immune system, is warding off viral infections. And so, again, that's another example of a prediction that's in print that is anticipating discoveries, and those predictions are flowing out of a commitment to a design, / creation model approach.
Amen. I think you nailed it there. Is the starting position is intelligent design and a creation, and these predictions naturally flow from that starting point. I find it interesting that I've heard a lot of anti-intelligent design people point out that intelligent design or the creation model doesn't make future testable predictions, and we know that would be the gold standard of science. But as you're pointing out here, no, it does. When we start from that position, we can make testable predictions that turn out to be successful.
Yes, and yeah, I want to be very careful as I say this, but oftentimes you hear this challenge leveled against intelligent design, against creationism, that if we embrace this idea that God did it, that it just would bring science to a screeching halt, that it they would stultify science. But the fact of the matter is, in my lifetime, I've seen, the evolutionary framework actually, in the case of identifying function for junk DNA, really stultify science. Because many people, again, I gave you the example of Craig Venter, and he wasn't unique in his views. Many people just didn't bother to study, the so-called junk DNA components of the genome. Why would you? If it's non-functional, why waste any time in research dollars that are so precious to study, something that you don't think is functional, right? And it was really not. It was really, in the context of looking at, other questions that people have stumbled upon function initially for junk DNA. Now, I think you can find a lot of people that are studying junk DNA with an eye towards anticipating function, but this is a relatively recent change in mindset. And so you could argue that this evolutionary view of the genome actually stultified scientific advance, it delayed scientific advance.
If you anticipate treasure, then you're going to go into these studies looking for essentially treasure and function. And one thing I wanted to point out that you said that was perfect was because I constantly hear the critics say that we are misrepresenting them because they never not predicted everything would be junk. They predicted function. But I think you nailed it perfectly when you said, sure, they predicted some function, but not the level of function that we are discovering in terms of the genome. And I think that's the perfect response to these critics that put forth that argument.
And let me say this, I've never seen anybody produce a citation where there is that prediction from an evolutionary biologist that junk DNA would be functional. I'm not saying that people didn't make that prediction. I've yet to see that I cannot find those kind of predictions. Now, if somebody can provide me with the references, I will be happy to stand corrected on that.
I've looked for that too, and I've not been able to find it. Not that it doesn't exist, I just haven't been able to find it. I definitely want to get this question in here. This is one of my own questions, and we got a ton of questions from the audience as well, because it's just such an interesting topic. So you said, Dr. Rana, in your article titled "Functional Pseudogenes Are Everywhere," and I highly recommend people check that out. So you said proponents of the evolutionary paradigm offer this rejoinder. Yes, some pseudogenes are functional, but the vast majority lacks function. The vast majority is junk. The question is this true? Is pseudogene functionality actually rare and thus still support for common descent?
Yeah, and what we're discovering, again, with the competitive endogenous RNA hypothesis is that unitary pseudogenes and duplicated pseudogenes are playing a role in gene regulation, where what we're learning is that genes produce transcripts, and those transcripts actually form kind of a family of transcripts that all are regulated by the same biochemical machinery. There are these enzymes that will essentially break down RNA as soon as it's produced. We used to think that gene regulation was only at the point where messenger RNA was produced, that you either allowed messenger RNA to be produced or you shut it or you inhibited its production. And that's how you would regulate gene expression. And there might be some translational control as well. But what we're now discovering is that there are these enzyme complexes that will break down RNA, and that this is setting up what biochemists call a feudal cycle, where messenger RNA is transcribed and it's broken down and it's transcribed. So in order to elevate the expression of a gene, not only do you have to produce messenger RNA, but you also have to downregulate the destruction of that RNA as well. And it turns out that what's happening is that you have these messenger RNA transcripts that are in effect part of the same family. They are regulated collectively up and down because they have the same regulatory sequences that are recognized by these enzyme complexes that break down RNA. And so what happens is that with regard to like pseudogenes and their corresponding coding, brothers and sisters, is that because the pseudogene has sequence similarities to the gene that is encoded, when its messenger RNA transcript is produced, it actually functions as a decoy, where this messenger RNA degradation machinery will actually break down the decoy or the pseudogene RNA, allowing the coding RNA to actually be translated. And this also explains, again, the function of unitary pseudogenes as well, because they are part of these gene networks or these transcriptional networks. And they can, again, function as a decoy for genes that are part of the network that actually are coding, again, functional proteins. So we have this really elegant theory that explains, again, the functional role of pseudogenes, both unitary and duplicated, and also explains why these sequences look like like gene sequences. And so this is an argument for widespread functionality, to pseudogenes. Now, another criticism would be, well, this is in reference to those pseudogenes that are being transcribed, but for those that are untranscribed, there doesn't seem to be any kind of role. This doesn't apply to those. Well, the fact of the matter is that, again, as I mentioned, there's some very real concern that's being expressed now that maybe pseudogenes are actually being transcribed at a higher level than we thought, because of the similarity between pseudogenes and, again, those genes that would code for functional protein products. Because of the similarity in sequences, many times people are actually detecting pseudogenes transcripts, and they are ascribing them to the transcripts of their functional counterpart or their coding counterpart.
Well, that's a great, thorough answer, Dr. Rana, and it's so important because we know over the years this has been one of the evolutionists' favorite argument, and rightfully so. I mean, if these really are shared genetic mistakes and they're widespread and they're shared in the same genomic locations as chimpanzees, who they say are our closest cousin, that certainly would be something we should take seriously. And the genome-wide functionality that you're pointing out really is fascinating. So my next question then, Dr. Rana, would be, I have also heard this same thing with endogenous retroviruses. Even though we have discovered ERV function in cell stress responses, immune responses, embryological development, gene expression, I believe, they will still say, similar to the pseudogene argument, that there are only a few, and therefore the best explanation for the function would be co-option. Is this really true? Is this also a rare occurrence, like they say with the pseudogenes, and how to best respond?
Yeah, and I think that at this point, to be fair, the jury is still out, because we are in the stage where we are beginning to discover, again, function for endogenous retroviruses, and we're beginning to learn, what that function is and how these sequences are functioning in an anti-viral capacity. So we're still, again, in the very nascent stages of making sense of this. So I think it's premature for anybody to draw any kind of conclusions about really the prevalence of functionality. And there was a study done recently looking at, a particular class of endogenous retroviral sequences in the human genome. And it turns out that these sequences are functional, but they are only transcribed in the very early stages of embryonic development. And they're playing an antiviral role, protecting a very vulnerable embryo from viral invasion, but they also seem to be playing a regulatory role where they are activating certain genetic programs that are necessary for the early stages of embryonic development in the process of cell migration and in cell differentiation. But the point is that if you studied those sequences in cells from adults, you would probably conclude that these sequences were not functional because they don't seem to be active at that stage in development, right? Or once development is completed. And so we have to be careful about necessarily declaring something to be non-functional just because we are studying cells at a particular stage of development or cells at a particular stage in the cell cycle. It could very well be that these sequences are functional only for a very limited time during the biology of the organism. And so we want to be cautious, I think, about declaring things. But to be fair, we're really at the beginning stages of recognizing function in ERV sequences. But this is a place where we would make a prediction, right? Is this an example of a rare occurrence of co-option, or is this a widespread phenomenon? If a design proponent or a creationist would say would predict that we should see extensive examples of function in EVEs and ERVs, whereas somebody that would advocate for the evolutionary paradigm would probably predict the opposite.
Amen. Amen. Well said. You nailed it when you said that it would be premature, because we would all agree that we are in the infancy of understanding the totality of the DNA language. And I think that's why us who hold to intelligent design and creation are confident enough to make these types of predictions. And I've seen you in your articles over and over again, Dr. Rana, make these predictions, functional related predictions when it comes to pseudogenes, ERVs, and so on and so forth. I wanted to ask you this, because this is another good article that you wrote. And I wanted to see if you could talk about the article's titled Endogenous Retroviruses Protect Early Stage. No, actually, that's one we kind of just talked about. This one is koala endogenous retroviruses protect against retroviral infections. Can you talk a bit about this and how it applies to both the creation and evolution debate?
Yeah, well, again, the significant aspect of that article is that, again, there are these retroviral sequences in the koala genome that look as if they are actually able to disrupt the process of retroviral insertion, as part of the retroviral replication cycle. Retroviruses have RNA as their genetic material, and they are reverse transcribed into DNA, and that DNA can become incorporated into the genome. And but endogenous retroviruses, at least those that are transcribed, are mobile elements. They're retrotransposons, where they can actually move or make copies of themselves and insert throughout the genome. They can move around, and it turns out that mobility, that retrotranspositioning that ERV sequences in koalas can undergo actually disrupts the incorporation of endogenous retroviral DNA into the genome. And so this is, again, identifying function for endogenous retroviral sequences, but also is explaining why they look the way they look. That the similarity to retroviral genetic material makes sense because of the nature of their functional mechanism. So it really ticks off a couple of boxes in terms of what is demanded of a creation model or an ID explanation, in terms of accounting for the presence of these materials in genomes.
Right. Another great response, another great article. I got those mixed up because I've read so many of your articles in the last couple of days. Great articles, great information. And the question I would then have, Dr. Rana, based on what you're saying, and there's still so much we have to learn about genome functionality, including the possible functions of ERVs, is the appropriate question to be asking then is, are these really the ancient remnants of past viral infections, or are they created units of DNA function? and thus we predict function.
Yeah, well, again, from an evolutionary perspective, you would argue that these are the vestiges of ancient infections. That would be the interpretation, where again there was co-option in a process of neo-functionalization. But until recently, the junk DNA argument, I think, really only made sense, or the presence of junk DNA in the genome really only makes sense in an evolutionary framework. And what we're seeing now is that somebody could legitimately view these sequences as part of the creator's handiwork. You could legitimately look at genomes as being, the work of an intelligent agent, as designed systems, because, again, we're making these kind of discoveries. So I think it's an important point to say that these discoveries aren't necessarily thoroughly undermining an evolutionary paradigm, but they're raising, I think, some sticky questions, for the evolutionary paradigm. But really the significance of these discoveries is that it really makes it, I think, thoroughly legitimate now to view genomes as, as designed systems. And the interesting thing is that the more that we continue to learn about genomes, the more and more sophisticated these systems seemingly become. They're not becoming less sophisticated, but they're really beginning to become more sophisticated and more elegant systems.
Right. I mean, now we're finding out that there's multiple functions. For example, protein moonlighting, we're finding that the genome's multidimensional, it's nested. We have multiple overlapping codes. All these things, from my understanding, were not really expected 50 years ago. They weren't predicted 50 years ago. They had that paradigm where it was one gene, one protein, one function.
Yeah.
And we find Yeah, go ahead.
Well, and something else that I think is important to recognize is that, a lot of the sequences in the genome are actually playing a role in establishing the three-dimensional structure of the genome. This is something, as you're mentioning, Donnie, is a relatively new insight, that there's a three-dimensionality to the genome, and that three-dimensional structure is really critical in terms of the function of the genome, because it's bringing enhancer sequences that are at a vast distance from the genome that they regulate, or from the gene that they regulate. It's bringing them in close proximity to the gene. But there's other things that these so-called junk DNA sequences are doing. Some of the pseudogenes that don't appear to be transcribed are playing a role in chromosomal stability, for example. There's a proposal on the table that maybe some of the so-called junk DNA sequences are serving as a mutational buffer, that mutations accrue at a particular rate. And if you just assume that the mutations are taking place randomly, then you need a certain proportion of sequences that are buffer sequences that can absorb mutations without compromising functionality. And so that's a model that's being proposed. There's what's called a nucleus skeletal role, where it turns out that the ratio of the volume of the nucleus to the overall cell volume has to be tightly regulated. And if the cell gets larger, the nucleus has to get larger as well. And the way that you regulate nuclear volume is through the DNA content in the nucleus. And so there are some people that argue that there's a nuclear skeletal role that's being played by junk DNA sequences that are creating just simply enough volume in the DNA molecule to force the nucleus to be a particular size, which allows the support of the operations in a larger cell compared to a smaller cell. So these are, again, other functions that people have identified that look like they're valid functions that also allow for the possibility that that the functionality may be simply structural in nature. It doesn't have to be necessarily in terms of regulating gene activity or encoding, functional, information.
Right. So many good points there, Dr. Rana. That's amazing. Each one of those points you brought up could probably take an hour lecture just to kind of go over all the details. Here's something that comes to my mind as you mention all this, because I've heard the critics, and I'm curious as to your thoughts. I've heard them attempt to undermine this widespread functionality of the genome, specifically the human genome, because we've tested so much of it, by saying, well, we've done all these knockout tests, genetic knockout tests with mice, and we find that they still grow up, they have babies, they live their lives, therefore most of the genome really is non-functional. Have you heard that response, and what's a good response to that?
I have, and that represents an interesting challenge, to this idea of functionality. But it also could be that there is redundancy that's in the human genome or in the genome of organisms that is not being accounted for in these experiments. And so, if you think about systems that are designed, that are well designed, many times there are redundancies that are built into those systems, particularly with respect to key elements of the systems that are playing a critical or a vital role in functionality. And so, it could very well be that, that there's redundancy again in the genome, or it doesn't have to be redundancy in the sense that there's just simply duplicated function, but it could be that the redundancy is such that there's other mechanisms that would compensate for the loss of function. And so that I don't think is factored into these types of experiments necessarily.
Amen. Great response. And I've even read that many non-coding RNA genes, even pseudogenes that we've spent some time on, are only expressed under certain conditions. So if some of these genes are being knocked out, and those genes being knocked out are not even expressed, or they're turned off because they haven't had the chance to turn on, maybe even through epigenetic means, then of course, the mouse is going to be fine at that point. So I better get to some of these questions too that are coming in from the audience, because I could ask you questions all day, Dr. Rana. You're a wealth of information. I appreciate that. So here's some good questions that have come in from the chat. Here's one from Logical, Plausible, Probable. It comes in the form of a super chat. So I appreciate that, John. John has interviewed you as well, Dr. Rana, on abiogenesis. I've linked that in the description box for people to check out. That was a great chat. So his question is, can you address the duplicate regions and how they are used based on cell type, efficiency versus waste / bad design? He says the usual Evo atheist talking point.
Gosh, I guess I'm not quite sure I get what logical, plausible, probable is getting at. I'm really sorry. I'm not sure what's being asked there. I'm sorry. I think in some respects, we maybe touched upon that when we talked a bit about redundancy.
Right.
That redundancy isn't necessarily a waste, but rather it makes the system more robust. So if that's what logical, plausible, probable is getting at, then I, but I'm not, again, I'm very, very sorry. I don't.
No, and that's, and if John, if you can expand, because it could also mean too, because from my understanding, they'll look to neo-functionalization, where you get a duplicate gene, which remains, I guess, useless for a while, and then eventually it's subjected to a series of mutations that makes it functional. I'm not 100% certain either, so maybe we'll wait for him to address. But if it does have to do with that as well, we've talked about neo-functionalization. So I guess the next question has to do with, this question comes in from Doki Doki SFT Fan Club. And Doki asks, is the gene desert paradigm going extinct?
Yeah, I mean, yes and no, I guess. I mean, there are vast regions of the genome that don't contain, sequences that would code for functional protein products or functional RNA products. Again, like ribosomal RNA or transfer RNA, things like that. But what's being replaced with the gene desert is that these regions appear to be, again, functional, that they very well may be playing some kind of regulatory role. So I think the answer is yes and no, right? You can have gene deserts now, but the implication in the past was that those are just vast regions that were non-functional, and I don't think we can make that statement now. Something that this brings up is that a lot of times people are declaring regions of the genome to be non-functional based, again, on this expectation that flows out of the evolutionary paradigm. They've not actually gone in and done experiments that demonstrate that those regions are non-functional. And so a lot of this is an inference that's being drawn, not based on experimental data, but really based on what the theory would entail. And so we want to be really careful, before we start making declarations, about whether or not parts of the genome are functional or not. There's a difference between, and even like with regard to the mechanisms for how so-called junk DNA sequences actually originate in the genome. These are mechanisms that are not unreasonable, but these mechanisms are really inferences that are being drawn based on the structural features that you look at, or based on some things that we know about the behavior of retroviruses and things like that. We've not actually necessarily directly observed these mechanisms in action generating, again, non-functional elements in the genome.
Amen. Well said. And it makes me think about the redundancy that you talked about earlier, and how redundancy is evidence for good design. It's evidence for forward thinking. But the evolutionist who holds to common descent, they go into it with assuming that redundancy is bad design or evidence of evolutionary leftovers. For example, I think about, the spare tire in the back of a car. To the evolutionist, that might be useless or wasteful. But if I enter an environment where I get a flat tire, it's a good thing I have that spare tire. It's redundant, it's necessary, and now it's going to help me out. It seems that a lot of the genome is indeed, as you pointed out, redundant for beneficial purposes. So I've got this from logical, plausible, probable. So he says, kind of expanding on that question, Dr. Rana, he says the evolutionists claim you wouldn't have duplicate sequences if it was actually designed, avoiding the higher expression levels of specific genes based on cell type. So I'm not sure if that helps, but maybe they look to duplicate sequences as you pointed out, redundant. It's there. It's right. Not really doing anything.
Well, something else that I'm beginning to see too now is this idea that the complexity in the genome is unnecessary, right? So it's arguing that maybe there's functionality here, but that functionality is unduly complex. So this is another challenge that's being raised to the design argument that I think relates to the topic today. And again, redundancy would be something that would fall under that category. Well, to have redundant elements would be unnecessary, if you've got a functional sequence. But as you're pointing out, that redundancy, particularly if it's in reference to a critical system, and that redundancy doesn't necessarily have to be just simply duplicated sequences. It could be that the functionality represented by that region of the genome could be taken up by other aspects of the organism's biochemistry, but there's a redundancy in the system so that if some functionality is lost, there's a compensation that takes place, or again, a redundant element that could simply replace it. So I think logical, plausible, probable is actually raising a really important point that again, we would see redundancy and duplication as essentially not a flawed system, but a robust system.
Amen. Well said again. I wonder, are you familiar with it? There's a paper, one of my favorite papers has to do with translational pausing and how a lot of these redundant elements, and I think specifically with third codon variation, third position codon variation, where I've heard a lot of critics say that they can use this because it's neutral variation, it's redundant, it's not really doing anything, and they can build phylogenetic trees, look at these nested patterns, and therefore conclude humans and chimps are related. But now we're finding out these redundant elements are not only there to correct error, but they're also involved in information flow. So we have multiple functions. And are you familiar with that, Dr. Anna?
I am, yeah. And and for example, redundancy in the human or in the genetic code actually produces optimization that minimizes error that could or minimizes the loss of information that could occur as a result of both substitution and framework mutations. And so this is an instance that's related to your point, where redundancy in the system is actually producing an optimal system, and this idea of synonymous mutations.
Yes.
We're learning that these, where the mutation produces the same amino acid, we're learning that this isn't, again, these synonymous mutations are not actually synonymous, because different codons actually can control the rate of translation, as you're talking about, pausing at the ribosome. And that impacts the folding of the protein, which really produces a different type of functional protein, or a protein with modified function. So, this is really a profound point, because a lot of the theory related to molecular evolution is built upon the idea of synonymous and non-synonymous mutations. And if synonymous mutations really are not synonymous, that has catastrophic implications for molecular evolution.
Amen. So much great information, Dr. Rana. It's so true. That really is catastrophic, because there are specific evolutionary biologists that I've even engaged that thought, or that have pointed to this neutral variation, synonymous codons, and therefore the hierarchies that result from it. They have pretty well looked at that as just a absolute evidence for ancestry. And now it seems like, are we looking at neutral variation or functional variation? Yeah. And therefore, would you say these hierarchies are then there by design?
Yes, I would. And the thing is with nested hierarchies, again, where you have these sequences that are groups within groups within groups that presumably reflect an evolutionary history. But if you think about it, let's use the automobile as an example. Nested hierarchies are very much part of systems that are designed, particularly when those designs have a history where the design has undergone an evolution, if you will. And so, like, if you think about automobiles and the very first automobiles, clearly there are certain design elements there that all automobiles share, right? But then as we look at future generations of automobiles, there might be new design elements and new functionality that's introduced into the automobile, and that then becomes part of the standard automobile design. And so, future generations beyond that point, we're going to have both of those design features as part of as part of their design. And then later on, when there's a third design element that's introduced that then becomes mainstay, you now have three design elements that, if you looked at all the automobiles through that history of automobile design, you would actually group those automobiles within groups within groups within groups within a nested hierarchy. And so, nested hierarchies are found in human designs all the time. So it doesn't necessarily demand evolution as the only explanation. It's consistent with an evolutionary history, but it isn't exclusive to an evolutionary history. It could reflect the design history, which is what we're talking about, right? When we look at the history of life on Earth, it's a design history.
Well, that's a fantastic point, Dr. Rana, because I find it interesting that when we start from the Bible, we know that we're made in God's image. So therefore, there's something about us that reflects the divine. And yet we build, as you pointed out, we build systems in these nested hierarchical patterns. And yet we see nested patterns in the biological world as well. It's almost like we're getting an idea or a sense into how God designed based on the way we design.
Yeah.
We're made in his image.
Well, it's interesting to me because, so oftentimes the argument is that shared designs, whether the designs are in the form of a nested hierarchy or some other type of pattern, is evidence for an evolutionary history, right? And so you could also argue, well, it reflects common design, as opposed to common descent. And oftentimes I hear skeptics say, well, why would an infinite creator limit himself to just certain design features, right? Why would he limit himself to these archetypical designs that might be varied but still are archetypical designs? Wouldn't a creator who has limitless resources produce an incredible diversity of life on Earth, where all the different life forms look different? Well, the thing that's, I think, missing in that analysis is the fact that because of shared design features and because of these nested hierarchies, what we learn when we study one organism actually can be generalized and applied to a large number of organisms. And this allows us to study model organisms and from that develop general principles about biochemistry and biology. And so in a sense, the shared features that we see in biology reflect a design that makes biology even possible, as a scientific discipline. You could argue that that biology has been designed for discovery, designed to make it amenable to learn and make sense of it very readily and very easily. And so I see that as part of God's providential care. And so there's an elegant reason why a creator would create things with shared features and with nested hierarchies, because again, it lends itself to discoverability.
Amen. Well said, Dr. Ron. It makes me excited because it's a great time to be a creationist, to be an intelligent design proponent, because of the models and the possible predictions and discoveries, as you've pointed out with discovery. So great response. As we kind of wind down here, because time really has flown by with you and we're going on an hour and a half, I could certainly talk to you about these things all day, Dr. Rana. I want to get this one in. This is another super chat that comes in from Logical, Plausible, Probable. So again, brother, I appreciate the support. He says, some pseudogenes are shown to keep enzymes from reacting until they reach the correct destination. His question is, can you explain this and how it's a problem or might be a problem for common descent?
This is, I have not heard of this functionality for pseudogenes. I'm not, and so I can't really comment, but this I think exemplifies the challenge that's in front of us. It's becoming very hard to actually catalog the functionality for so-called junk DNA elements. I mean, it's almost like we're in an avalanche of discoveries that are taking place right now, identifying function for the junk DNA elements, or the so-called junk DNA elements, not just pseudogenes and ERVs, but other sequences like line and sign sequences. And so I can't comment on it. I'm going to have to. So thank you, Logical, Plausible, Probable, for pointing that out. I'm going to have to dig into this myself. But this is actually a good problem that it's really challenging to keep abreast of the discoveries that are identifying function. And again, this problem is something that is endemic to a ID or creation model approach. It's not something you would expect from within the evolutionary paradigm.
Amen. Amen. It's a good problem to have, that it's hard to keep up on all the incoming discoveries and data that ultimately supports our position and our position of design rather than descent. As we kind of wind down here, Dr. Ryan, I was wondering if you could talk about an important class of RNA molecules called MicroRNAs, and you've written about them in a couple of the articles that I've read, and I think it's a fascinating topic.
Yeah, well, one of the things that we're discovering is that, there is, the so-called dark matter of the genome, where you have all of these regions that are transcribed into what now is becoming like a zoo of RNA molecules that are of different sizes, that are playing regulatory roles. And microRNAs are in that category, just like long non-coding RNAs. And they all are playing, again, roles in gene regulation, and they're playing regulatory roles, and it, again, this is something that is, kind of a revolution, if you will, in molecular biology, is that there are all these RNA molecules that again are playing vital roles. And this goes again to the point that we were making very early on, is that you can't just simply assume that because something is transcribed that it's noise. Because these microRNAs and again the long non-coding RNAs would be included in the noise, at least with the original assessment of the ENCODE results. And again, we're discovering that these things are playing a vital role. So, we're in our infancy, and we're discovering things about the genome and its structure and function that are just highlighting the stark complexity, but yet it's also a sophisticated and elegant complexity that we're just beginning to grasp. And to me, it's just the genome is becoming an incredibly marvelous biochemical system.
Amen. And from my understanding, we're just beginning, Dr. Rana, to understand just how all the different windows and stages of the developmental process works, and how the coding and non-coding DNA portions of the genome interact, and in a way that coordinates how these structures are sequentially put into place to produce a fully functional organism. And therefore, it really is premature for the evolutionists to say that a lot of the genome is just spurious or junk or useless, because to me, and I wonder if you have these thoughts as well, a lot of the non-coding regions are probably involved in these various developmental windows.
Oh yeah, I think that's absolutely true. And it could very well be that, again, there are portions of the genome that are only active at certain points in development. And it's very difficult for us to assess that right now when it comes to the early stages of development, right? Because you're having to work in vitro on human embryo systems, and that opens up a whole can of worms, right? But the fact of the matter is, it's very difficult at this point to truly get an assessment. But I suspect that very much is the case, and I think one of the things that is going to be something I predict happens as we move into the future, and to some degree it already is that the regulation that's taking place in the human genome, and again the genome of other organisms, is so sophisticated and complex that it's really going to take a systems engineering approach to really figure out what's going on in terms of regulation. And I think we also are going to discover that not only is systems engineering critical to interpret the regulatory networks that are involved in regulating, the activity of the human genome, that I suspect we're going to discover regulatory systems that inspire new advances in systems engineering as well. That we're going to learn things from the human genome that are going to open up new vistas in that area of engineering. So that's another prediction that I think we could make, that I'm not sure an evolutionary biologist could make the comparable prediction.
Amen. Well said. And I would agree that is a good prediction, and where we are heading, that's probably going to be confirmed. So I'm going to end it with this last question. This is a question that kind of came in all the way at the beginning to respect redefined living. I want to get it in there. Hour and a half really has flown by. So much great information, Dr. Rana. So the question is, when you say information, is that analogous or literal when concerning the genome?
I think the information is literal information. The information in DNA is digital information. There's discrete units that are reflected in the nucleotide sequences, and when we're talking about a gene, really in the codons, the coding triplets. But in addition to that, we're now learning, as we talked about, that there's a three-dimensional structure, to the genome, and that three-dimensional structure could be viewed as being analog information. So there's really both digital and analog information embedded within the genome, where digital information is discrete and analog information is continuous. And so when you talk about three-dimensional shapes, there is a continuum that those shapes are on, and so those shapes are actually have information instantiated in them, and so I think it's appropriate to think of the genome as having both digital and analog information. So it's an incredibly sophisticated system in that respect. And again, we're just beginning to understand how to decode that information. So, what's remarkable is that embedded in the sequence of DNA is not just simply, the genetic code, which defines the information, but there is a histone binding code. There is a transcription factor binding code. There is a spliceosome code that are all overlapped and stacked on top of each other in the same sequence. So you have multiple overlapping codes that are taking the same sequence of nucleotides and communicating different sets of information to different aspects of the cell's machinery. That to me is mind-blowing. And in fact, the genetic code that we see in nature, which is in effect universal, is redundant, and that redundancy allows for multiple overlapping codes. And the genetic code in nature seems to be optimized among random codes that could be imagined to harbor multiple overlapping codes. So, the information that's embedded in DNA is so complex and so sophisticated and so elegant. I think an information scientist would struggle to produce a system that could harbor multiple overlapping codes at the same time.
Wow, great response. It really is science fiction what we're seeing on the molecular level. It reminds me, what you said reminds me of Michael Behe's book Darwin Devolves, with how sophisticated and complicated the genome is, and yet evolutionists want to say that mutations, which I understand are mostly damaging and functionally compromising, could take a single-celled like ancestor into, let's say, a whale over time. When with such a system that's so complicated, it's more likely that mutations are going to damage or degrade functionality rather than improve it or make something better.
Yes, because that's true. I mean, when you have highly optimized systems, any kind of change to that system is going to move you off of that optima, right? And so I think that's very much the case. But what's also remarkable is that mutations are a consequence of the fact that we live in a world that conforms to the laws of thermodynamics. And yet what we see, in the cell is all these mechanisms that are put in place that actually turn what could otherwise be deleterious mutations into neutral mutations or mutations of minimal consequence. And so there are all these redundancies that actually create robustness against mutations. And this is in the nature of the genetic code itself. It's within the fact that there may be sequences that are serving as mutational buffers. It's in the redundancy that we see within the system. And so plus, not to mention DNA repair mechanisms that are in operation as well, which is in and of itself a whole nother facet of the design argument. And so, so the remarkable thing is how few mutations actually compromise function. In light of the fact that these systems that we're looking at are so highly optimized.
Well, like you said, we've got these DNA repair enzymes and systems that help repair error. And if we didn't have those, we'd probably be extinct in a generation. I mean, that's thinking ahead. That's forward thinking, which points us back to a forward thinker. And the buffering makes me think of your car, the treading on your tires. You know, over time that slowly wears down, but it does take time, and it's there to prevent the the ruining, I guess, of your tires after one drive. You got a lot of buffer there that's designed that way, just like the genetic system. So great points, Dr. Rana. I want to hand it to you. We're going on an hour and 40 minutes. We still got a great chat. So much great information. We touched on so many good topics: pseudogenes, hierarchies, endogenous retroviruses, junk DNA, gene deserts. So a great, thorough show. I want to thank you so much for giving us your time, for giving me your time for this important program. I know you're busy, Dr. Rana. So I wanted to hand it to you for some final words, final thoughts before we close it down.
Yeah, to me, I think we're at an exceptional time in scientific history when it comes to genomics, and it's remarkable to me to think that it was about 20 years ago that the first draft of the human genome sequence was produced, and what we've learned in the contravening 20 years has been nothing short of absolutely astounding. Particularly for people that hold to an ID perspective or a creation model perspective. I suspect that the next couple of decades are going to unleash even more profound discoveries. But to me, the thing that's so encouraging is that the more that we're learning about the human genome and the genomes of other organisms, the more that we're seeing a case for design, that is a case for design that is mounting, and I think there's going to be a point in time when we look back and we could never imagine anybody thinking that the genomes of organisms were anything other than these incredibly elegantly designed systems that suggest the work of a mind.
Amen. Well said. Well said. People are in the chat are saying thank you for a great presentation. John did list the book that he was recommending to you, but the chat's moving too fast, and unfortunately I lost it. I apologize. John, maybe you can email it to me and I'll email it to Dr. Rana. We've had a great chat, great final words, Dr. Rana, and I can't thank you enough. So God bless you. God bless everybody in the chat. Please share this around. This content is important, and we'll see you guys later. God bless. Standing for Truth is out. Welcome to Standing for Truth. I am your host, Donnie, and it is a privilege to have Dr. Fuzz Rana back here with me today. Dr. Rana was here approximately 2 months ago for a presentation and discussion on junk DNA. Today, we continue that discussion with a strong focus on addressing the arguments that are being used to save the junk DNA paradigm. Dr. Rana, thank you again for being here and giving us your time for this important discussion.
Well, Donnie, thanks so much for having me. I really, really appreciate the opportunity and look forward to the time that we're going to hang out together.
I appreciate it. I'm extremely pumped for this. This is one of my favorite topics. So definitely this is going to be exciting. We've got a great audience, and we will be having questions from the audience as well, guys. So please make sure you are tagging me at Standing for Truth. We'll get to some of those questions after Dr. Rana's presentation. Now, there's also more information about Dr. Rana in the description box. I want to give him a brief introduction before we get into the presentation. Now, Dr. Rana is VP of Research and Apologetics at Reasons to Believe. Fuzz earned a BS in chemistry with highest honors at West Virginia State College and a PhD in chemistry with an emphasis in biochemistry at Ohio University. You can also find a lot more about Dr. Rana, as I pointed out, including many of his must-read articles in the description box of this video. So that being said, Fuzz, I think we're just going to get right into it. Unless there's something you wanted to add, we can kind of just get right into the presentation.
All right, well, it sounds good. Let me go ahead and share my screen and pull up the PowerPoint. Okay, and here we go.
There we go.
All right. And how does that look? Or is
Looks
it coming?
great.
All right,
Oh, yeah.
perfect.
Yeah, looks good.
Okay, so as Donnie said, I'm going to be taking a little bit of time here and just presenting some of my thoughts when it comes to the whole junk DNA uh, phenomena and whether or not junk DNA can be rightly understood as being consistent with a case for intelligent design or a case for creation. And as Donnie mentioned a couple of months ago, I was on Standing for Truth and presented some material there at that point in time. And so what I'm going to do is a quick reprise of some of the stuff that I presented, and then we're going to kind of get into some of the challenges that people have leveled against the case for creation, again, related to the junk DNA argument. And the first thing that I want to do is talk a little bit about why people think that junk DNA represents such a powerful case for biological evolution, for an evolutionary history of life on Earth. And there's really, in a sense, two broad reasons, from my perspective at least. The first has to do with something known as the C-value paradox. And the C-value refers to the amount of DNA that is found in the nucleus of the cells in an organism. And typically, the amount of DNA is consistent from cell type to cell type in an organism, but differs from organism to organism. And you can think of the C-value as roughly equivalent to genome size. And at one point in time, people naively thought that there should be a correlation between organismal complexity and the amount of DNA in the nucleus of their cells. But lo and behold, when you plot out the C-value as a function of the different types of organisms that are found in the living realm, we see no relationship whatsoever between organismal complexity and the amount of DNA in the genome. And the resolution to this is the C-value paradox, which argues that the amount of DNA in an organism's genome has no bearing on the functionality of that DNA, but rather it is the result of an accumulation of so-called junk DNA through a variety of different mechanisms that just happen to constitute an organism's genome. So the resolution to the C-value paradox is that most of the DNA in an organism's genome is non-functional junk DNA, and that is what explains the DNA content, not organismal complexity. And of course, when the human genome was sequenced, we discovered that the human genome harbors about 20,000 genes, which is a much smaller number than anybody would have anticipated, and that most of the human genome, somewhere between 95 to 98% of the human genome, consists of non-functional DNA. And in fact, when you look at the human genome, you can categorize the human genome based on different types of so-called junk DNA sequences, and so it looks as if you can explain the structure and the composition of the human genome through, again, the C-value paradox. And then, in addition to that, the fact that we can categorize the different non-functional DNA into different classes suggests to many people that we can understand this DNA again as the vestiges of an evolutionary history, where we understand mechanistically how these different types of junk DNA sequences are introduced into the human genome or the genomes of other organisms, and that we can actually organize the sequences for the different classes into these nested hierarchies, that these sequences are shared, and they also can be organized into nested hierarchies, which presumably reflect an evolutionary history. And so for many people, they see, again, junk DNA as really compelling evidence for biological evolution. It makes sense from an evolutionary paradigm, again, why the genomes of organisms contain so much junk and why that junk DNA again, has the distribution that it has among groups of organisms that naturally cluster together. And I would actually agree that, when you look at the case for biological evolution built upon, again, junk DNA, it on the surface is a very compelling case that can be made for an evolutionary history of life. But this leads me then to the next point that I want to make, and that is that one of the projects that I have at Reasons to Believe is to build a creation model for the origin and the design of genomes. I am of the conviction that there is a creator that is played a role in the origin and the basic design of life, as well as the history of life on Earth. And the reason that I have for that conviction has to do with challenges to accounting for the origin of life through chemical evolution, coupled with what I see to be an elegant design of biochemical systems. I also see elegant designs in biology. I think the evolutionary paradigm struggles to explain certain key transitions in the history of life. I won't get into those specifics. And so these are reasons why I think that an evolutionary framework is insufficient to account for the origin, the fundamental design, and even the history of life on Earth, at least in its totality. And again, I also see compelling reasons to think that there's a creator involved in the origin, design, and the history of life because of the elegant designs that I see. So the question then becomes, within that framework, is it possible to understand the human genome and the genomes of other organisms from a creation model perspective? And in order to do that, there are a couple of criteria that we have to meet. But before I get into that criteria, I just want to pause for a second and discuss a little bit about the influence of philosophy on the way in which we interpret biological phenomena. Because the framework that we see today for contemporary science, the philosophical framework, is methodological naturalism, namely that when we seek to explain phenomena in the universe and specifically phenomena within the biological sciences, we're looking for mechanistic, natural process, materialistic explanations. Any appeal to the work of a creator is excluded as a possible or a viable explanation a priori for philosophical reasons, not because that is what the evidence necessarily demands. And so within this framework of methodological naturalism, then when you look at the shared junk DNA sequences you see in genomes of organisms that naturally group together, it makes sense to think that these indeed must reflect an evolutionary history, because this is the only possible explanation that can exist. This is the only possible category of explanation that can exist. On the other hand, if we relax the requirements of methodological naturalism, we could understand that the shared features reflect common design, not common descent. And it's interesting to me that prior to Darwin's theory of evolution, the phenomena of homology was well known. And this is what we're talking about when we talk about shared biological features. These shared features are referred to as homologous structures. And Sir Richard Owen, who was a prominent biologist that preceded Darwin, was considered to be the father of comparative anatomy. He was viewed to be in the same category as an Isaac Newton in terms of his scientific prominence. And he had this idea that these shared features, these homologous structures, reflected an archetypical design that existed in the mind of the first cause that was then functionally manifested in the created order. And he conceived what he called the vertebrate archetype, that again was an idealized design that was then functionally manifested in the created order in a variety of different ways to produce a wide range of different types of biological systems and organisms. And Owen saw the archetype as being this the ultimate expression of design, because he marveled at the fact that there are these archetypes that exist that are so robust that they can be varied to produce a wide range of biological systems with a wide range of function. And so this is what Owen writes: the satisfaction felt by the rightly constituted mind must ever be great in recognizing the fitness of parts for their appropriate function. But when this fitness is gained, as in the great toe of the foot of man and the ostrich, by a structure which at the same time betokens harmonious concord with a common type, the prescient operations of the one cause of all organization become strikingly manifested to our limited intelligence. So in other words, the way Owen understood homologous structures is that these reflect common design, not common descent. And what Darwin came along when he proposed this theory of evolution was he co-opted Owen's idea of the archetype, but then he evolutionized it to be a hypothetical common ancestor. And so what we see with again these shared features is that there are two different ways that they could legitimately be interpreted. Both ways actually have a precedence in the history of biology: common design or common descent. And so what we're doing when we present a creation model for the structure of genomes is that we're looking at genetic archetypes, or we would argue that the shared genetic features reflect archetypical designs that then are manifested in a wide range of different organisms, reflecting common design. Now, there's another piece of philosophical there's another piece to the philosophical influence on interpreting the biological record, and this has to do with something called the underdetermination problem. That is, it's this recognition that theories are underdetermined by data, meaning that the same set of data could equally be appropriated by two radically different types of theories and satisfy the requirements of both theories, provide support for both theories. And so I would argue that common design and common descent, given this underdetermination problem, are both equally valid ways of interpreting shared genetic features that we see in organisms, with the idea that, again, if we relax the requirements of methodological naturalism, I think the idea of common design is an equivalent theory to common descent. Now, of course, if we're going to adopt that approach, then we need to have, from a creation model perspective, a rationale for why there are junk DNA sequences in genomes, but also we have to have an ability to explain the similarities between these junk DNA sequences and the types of sequences that would be produced by a variety of mechanisms that evolutionary biologists think have generated these different types of junk DNA sequences. As an example, if we're going to argue that endogenous viral elements and endogenous retroviruses in genomes of organisms are functional, then we need to not only explain how they are functional, but also why they bear a similarity to viral genetic material. Or if we're going to say pseudogenes are functional and they are intentionally introduced into the genome as the creator's handiwork, then why do they have the appearance of, or similar appearance to, genes? These are things that we have to do if we're going to advance, again, a creation model. Now, as we talked last time, the ENCODE project really goes a long way towards satisfying the first criteria. Namely, the ENCODE project demonstrated that a significant proportion of the human genome consists of functional DNA elements, or at least what the ENCODE project interpreted as functional DNA elements. And the ENCODE project performed a number of assays to identify functional sequences in the human genome. This includes transcription, the binding of transcription factors, histone binding, the binding of modified histones, methylation, and then three-dimensional interactions in the genome. And through this work, Phase 2 of the project demonstrated that 80% of the human genome is functional, or at least displays biochemical activity that was interpreted initially as functional. And we now are at phase 4 of the ENCODE project, and phase 4 has not really produced a numerical quantitation of the percentage of the genome that's functional, but continues to affirm this original result, that a significant fraction of the human genome appears to play a functional role in genome biology. And so this, of course, is a radically different perspective on the genomes of organisms than we see when it comes to the evolutionary paradigm. And so instead of 5% of the human genome being functional, as a result of the ENCODE project, the initial result indicated 80% of it was functional, with the expectation that eventually, as we continue to do a deeper characterization, that number will go closer to 100%. Now, that's kind of a quick synopsis, a quick reprise of what we discussed the last time I was on the show. And now what we want to do is kind of get into a response that people have to the findings of the ENCODE project and the general case made by ID people and creationists with regard to, again, the functionality of junk DNA. And it's interesting to me that the results of the ENCODE project have ignited what Carl Zimmer called in an article published in the New York Times, the junk DNA wars. And this is a civil war that's taking place within biology. This is not a war taking place between creation model advocates and ID proponents and evolutionary biologists, but rather this is a civil war taking place between genomic scientists and evolutionary biologists, where genomic scientists, by and large, I think, accept the importance and the significance of the ENCODE project results and find that information useful, whereas evolutionary biologists have derided these results, arguing that, in effect, that if indeed the ENCODE project results are correct, then it means that key features of the evolutionary paradigm simply cannot be valid. And because, according to evolutionary biologists, we know that the evolutionary paradigm is airtight, it means that the results of the ENCODE project must be fundamentally flawed. And this is an interesting argument, I think, because it's somewhat of a bizarro world approach to science, where the theory is sitting in judgment of the data as opposed to the data sitting in judgment of the theory. And one of the chief critics of the ENCODE project is a biologist, I think at the University of Houston, by the name of Dan Grahr, who shortly after the publication of the ENCODE project results, published a paper in Genome Biology and Evolution, where he really took the ENCODE project and the results of the ENCODE project to task. And even though this article is somewhat dated now, it's not quite 10 years old, many of the ideas that Grahr presents in this article are ideas that are still in play in terms of criticizing and critiquing the ENCODE project. And so I'm going to step through some of Grahr's criticisms and then offer a response to them. And then from there, we'll continue to look at other types of critiques that have been leveled as well. In a nutshell, there are three major issues that Grahr and others like Grahr have identified with the ENCODE project. The first has to do with the definition of function. The second has to do with the logic of how the ENCODE project established functionality. And then the third has to do with what Grahr claims is a confusion between biochemical activity and function. Now, with respect to the definition of function, Grahr argues that there are two different fundamentally distinct approaches in biology to function. One is what he calls the selected effect. The other is what he calls a causal function definition. A selected effect definition, according to Grar, refers to a trait for which it has been selected or by which it has been maintained through, again, through natural selection, through selection. So in other words, what Grar is basically saying is that for something to be, again, functional, you have to understand that functional element in evolutionary terms. And if it shows evidence that that region of the genome has been selected, that it's under the influence of natural selection, then we can consider it to be functional. Otherwise, we can't legitimately consider it to be a functional piece of DNA. According to Grar, the causal function concept is that for a trait Q to have a causal function G, it is necessary and sufficient that Q performs G. So in other words, if we say, for example, that a particular region of the genome is a protein binding region, let's say it binds a transcription factor, that binding of the transcription factor would be the functional role, then it's necessary and sufficient for, again, that Q performs G. So in other words, if you observe that if a piece of DNA is functioning as a transcription factor binding site, and we observe that it binds a transcription factor, then we can conclude that it must be functional in serving a role as a transcription factor binding site. That's the causal function definition. And according to Grar, only sequences that can be shown to be under selection can be claimed with any degree of confidence to be functional. Now, the first point I have to that assertion on the part of Grar, because when you adopt that approach, then you would argue that minimally about 10% of the human genome, maximally about 10% of the human genome is functional. The rest is non-functional DNA, according to that definition. But the first point I want to make is that that's a theory-laden definition of function. In other words, that's a definition of function that is predicated on the evolutionary paradigm being valid. And whether you believe the evolutionary paradigm is valid or not, at this point, that's irrelevant. The point is that any kind of definition of function really should be a definition that is independent of any theoretical framework, of any paradigmatic framework. Because as soon as you have a theory-laden definition of function, that again, I think creates a circumstance in which it becomes impossible in principle to actually now evaluate that theory in light of that particular observation. So this is not this is a problematic. This is, to me, is problematic with Grar's assertion that function is only for regions of the genome under selection. Now, I would also say that when you go to any other area of science outside of biology, science is about establishing cause and effect relationships. That causal function concept is essentially, it essentially permeates all other disciplines of science. That's what science is about, is trying to establish those causal relationships and employing a causal function definition of functionality. And in fact, as a biochemist, almost every study that I've ever done involves that approach to functionality, namely a causal function approach. And so, again, this is the point that I just made, that causal function is routinely used in biochemistry and other areas of science. Now, in preparation for the show, as well as in an interaction I had on social media, both Donnie and another person I was engaging brought up this particular article, this perspectives article published in PNAS in 2014 as essentially an article that explored the question, how should function be defined in biology? And they brought this article up as a challenge to the ENCODE project's approach to defining function. However, when I read the article, I found it to be a very thoughtful, very balanced, and very, again, insightful piece on what does function actually mean in a biological context. And what they identified in this article were 3 different approaches to function: an evolutionary approach, which is similar to the approach that Gar is arguing for, namely that regions of the genome are functional if they're under selective constraints. They also brought up the genetic approach to function, which is to argue that if you have a perturbation in the genome, you look at to see what is the phenotypic effect. If there is a significant phenotypic effect, then that you would consider that particular region of the genome to be functional. And then the last approach would be the biochemical approach, which is what the ENCODE project engaged in, was looking for different types of biochemical activity, where that activity was then assumed to indicate functionality. And what you see is that the biochemical approach tends to give you a very high degree of functionality when you look at the human genome. The evolutionary approach gives you a very low degree of functionality. And so these are, again, definitions that are, if you will, on the opposite ends of the extreme. And in the article, the team that was writing this piece essentially rightly pointed out that each of these approaches have limitations, and each of these approaches have strengths. And they actually argue for kind of a blended approach where all 3 different approaches are coming into play to assess functionality in the genome. But they highlight problems with each of these. For example, when it comes to, again, looking at regions of the genome as to whether or not they're under selective constraint, again, this is a theory-laden approach to defining function. But on top of that, this approach, I think, neglects the fact that that just because something isn't under selective constraint doesn't necessarily mean that it's still not performing a function. And so I don't think that this is actually a very good approach to defining function. And let me just give you kind of a silly example, because I could argue that my foot may not have anything to do with my capacity for reproductive, fitness, but yet, and you could cut off my foot, you could cut off both of my feet, and I would still have, again, the same reproductive fitness as I would if I had both functioning feet. At least, or I could imagine circumstances where that would be the case. Yet it would be inappropriate to say that my feet don't perform some kind of function. So that's, to me, again, a kind of a simplistic illustration, but I think it highlights the limitations of the selective constraint approach. When it comes to the phenotypic effect, this approach, again, is an interesting approach for function. But again, one of the problems here is that we don't understand the relationship between genotype and phenotype. And so until we have that understanding, it's very hard to use this approach, I think, to establish unequivocally whether or not regions of the genome have function. And if there is any kind of redundancy in terms of genomic function from region to region, this phenotypic effect is not going to you could disable a region of the genome and see no effect on the phenotype and conclude that that region is not functional. But if, again, that the functionality of that region is redundant, it's where it can be compensated for by other regions of the genome, you couldn't rightly say that that region was non-functional. And then last but not least is the biochemical approach. And one of the points that is made by these research teams is that this is actually a fairly robust approach to establishing function, because when you're measuring molecular scale activity, you're very close to the mechanism by which that region of the genome is actually operating. You're actually delving into the mechanistic operations, and so you're very close to having a fundamental understanding of the structure-function relationship between those regions of the genome and, again, their functionality. And so this is actually a robust way of gaining insight into function, much more so than the other two approaches, which give you no real insight mechanistically as to what is the basis for function. So I think the ENCODE project, in other words, made the right decision to use a biochemical approach, because it does give us information that leads to a mechanistic understanding, a fundamental understanding. Now, another criticism leveled by Gurar is the logic that the ENCODE project employed. He accused the ENCODE project of affirming the consequent, which is a logical fallacy. And so, in other words, if P then Q, we observe Q, therefore P must be true. This is called affirming the consequent. Now, I would argue that affirming the consequent is a problem if you're dealing in deductive reasoning. But in science, we're dealing in inductive reasoning. And in fact, we engage in this kind of reasoning all the time, because and it's not a logical fallacy to do that in science when you reason this way, because you're reasoning inductively, not deductively. And in fact, and so what Gurar would argue is this, that DNA segments with a particular function F display property P. Property P is observed, therefore the DNA segment has function F. So if we say that transcription factor binding sites will bind transcription factors, we observe transcription factors binding to a particular site, therefore we assume it's a transcription factor binding site. That would be, according to Gurar, affirming the consequent. But in inductive reasoning, that is actually a legitimate way to, again, think about phenomena, to work towards establishing cause and effect relationships. Because again, there's a difference between inductive and deductive reasoning. And in fact, nearly all scientific measurements ultimately affirm the consequent because they're done inductively. What that means, though, is, of course, that we have to do additional work to affirm what we think to be the case. So if we think a region of the genome is a transcription factor binding site because it binds transcription factors, we need to do additional experiments to confirm that result. But that type of reasoning is not a flawed reasoning. It's not flawed logic. The ENCODE project employing that type of reasoning was well-founded. And by the way, Gurar affirms the consequent when he argues for a selected effect definition, where he argues that if a region of the genome is under selection, it must be functional. We see a region of the genome under selection, therefore it's functional. That's affirming the consequent. And so Gurar is guilty of that same kind of logical fallacy as the ENCODE project is. Now, to me, the criticism that Gurar raises here is actually legitimate criticism, and that is the ENCODE project measured biochemical activity and assumed that activity meant that it was functional, that they ascribed activity or function to activity, to biochemical activity. And it's quite possible that some of that activity was simply spurious, that some of that activity was essentially biochemical noise. And I think this is something that is a legitimate concern that's being raised. However, when I saw Gurar raising this concern, almost 10 years ago now, My response at that time was that I was skeptical that that the activity measured by the ENCODE project was largely noise as opposed to reflecting functionality for two reasons: for energetic considerations, as well as for the concerns about whether or not you would have an interference problem if that level of activity was actually biochemical noise. And so let's kind of step through each of these in turn. So when it comes to transcription, I think about 60% of the genome, human genome, is transcribed, if I'm not mistaken. And if that's the case, and much of that transcriptional activity is biochemical noise, that to me would be deeply problematic, because transcription is a energy intensive, resource intensive process where you're utilizing essentially, nucleotides. You're utilizing nucleotides, which are valuable resources for the cell, to produce RNA molecules that again are just simply the result of biochemical noise. Plus, it takes 14 kilocalories per mole every time you add a nucleotide onto a growing polynucleotide chain, whether it's RNA or DNA. And that's a very energy intensive process. And so because you're using up valuable resources, because you're using up large amounts of cellular energy to produce transcripts, if they are non-functional, it seems to me that even from an evolutionary perspective, that kind of wasteful process would actually be minimized. There would be mechanisms in place and very strong selective effects in place that would essentially try to dampen that kind of unnecessary transcription. And it turns out that when we evaluate the transcripts, that many of these transcripts actually, again, of the 60% of the genome that are transcribed, seem to have functional utility. 2% of the transcripts are the protein coding genes, 27% would be regions that are transcribed but not translated, and these would be coding associated. So this would be things like introns, for example. There's also regions that are transcribed but not translated that are non-coding regions. And then pseudogenes are also transcribed as well. And when we look at the list of the transcripts, not only do we see, again, messenger RNAs that reflect protein coding regions of the genome, but we also see, transfer RNAs, ribosomal RNAs, nucleolar RNAs, spliceosomal RNAs, microRNAs, small interfering RNAs, and long non-coding RNA molecules. And all of these that we see are known to have function. In fact, a few years ago, a research team looking at non-coding RNAs basically argued that most transcripts play some kind of functional role. That non-coding RNAs are not simply transcriptional byproducts or splicing artifacts, but comprise a diverse population of actively synthesized and regulated RNA transcripts. These transcripts can and do function within the context of cellular homeostasis in human pathogenesis. And the point here that I'm making is this: that this is a research team that is working independently of the ENCODE project and has concluded that the transcription that we see in cells is actually mostly functional, that it's not biochemical noise, and that this is essentially giving legitimacy to the results of the ENCODE project. Now, this brings us to the point where I want to talk about the competitive endogenous RNA hypothesis. This is an idea that essentially explains the functional role of duplicated and unitary pseudogenes, but also explains the role that long non-coding RNAs play, as well as a role that microRNAs play also. And the idea behind this model, which is actually gaining some strong experimental support, is that RNAs actually are transcribed as families of RNA molecules. That transcripts form a family of transcripts, and there's different families of transcripts, and that these different families of transcripts essentially are regulated in concert with each other. And what defines a particular family of transcripts would be the microRNA binding sites on the transcripts. And so what happens is you have RNAs that are transcribed that have, again, the same microRNA binding sites. When these microRNAs bind, let me go back here. When these microRNAs bind to the transcripts, they essentially target those transcripts for being destroyed by the RISC complex. And so this destruction means that when RNAs are basically being transcribed, they also are being targeted for destruction. So it's kind of like a biochemical feudal cycle where RNA is being produced and then is being destroyed. And so this is actually a type of gene regulation. So it turns out that not only is gene regulation affected by whether or not a transcript is being produced, but also once it's produced by whether or not it's destroyed before it has a chance to be translated at the ribosome into a functional protein product. And so the idea here is that you have these, again, microRNA sites that create families of RNAs, families of transcripts. And so if you upregulate the transcription of one of members in that family, they essentially are going to function as molecular decoys that will lead to their destruction. And as a result of that, they will allow other members in that RNA family to actually be translated at high levels. And so this is a role that is being played by not only long non-coding RNAs, but also by unitary pseudogenes and by duplicated pseudogenes as well. And in fact, duplicated pseudogenes actually are playing a decoy role in terms of gene regulation. But the point here is that, again, you have these RNA families in which the translation of members in that family are dependent upon, again, the levels of other transcripts that are part of that family. And that you have, again, this network of RNA molecules that, again, are transcribed and translated in concert with one another, where this decoy mechanism is at play. And so this explains in part why you have long non-coding RNAs and other types of RNA molecules being transcribed. They're playing an important role in gene regulation. Now, another criticism that we see that's kind of related to this idea of transcription is that some of these transcripts are produced at very low levels and therefore, again, must be spurious, must be biochemical noise. But this is not necessarily the case, because just because a transcript is, again, being produced at low levels doesn't mean that it's actually not having a very important and very impactful regulatory effect. Just you can't necessarily correlate the level of transcription with whether or not it's functional or whether or not it's biochemical noise. Now, another concern I have with this idea that activity is not functionality, but it's biochemical noise, is that, again, if you're creating, this has to do with primarily the transcription factor binding sites, is that you would basically have a biochemical interference taking place if you're producing RNA transcripts that are randomly binding to sites in the genome. And so, for example, it's not unreasonable, I think, first of all, to ask the question, is transcription factor binding actually noise, or is it actually functionality, or displaying functionality? Because, most transcription factor binding sites are about 4 to 10 base pairs in size, and there's about, typically about 100 binding sites per genome for a particular transcription factor. But when we look at the genome, which is 3.2 billion base pairs in size, there could be anywhere from 1 million to 1 billion potential binding sites for that particular transcription factor, because you could statistically have that binding site recur by accident in a genome that's 3.2 billion base pairs in size. You could have variants of that transcription factor binding sequence that would be potentially high affinity binding sites, other variants that would be medium affinity, and then some variants that would be low affinity binding sites. But the point is that you could have, in principle, statistically speaking, a large number of potential binding sites. But a study done and published in 2016 indicated that high affinity non-functional binding sites in the human genome and the genomes of organisms really across the board are extremely rare. And in other words, they actually measured transcription factor binding sites affinities, and then through statistical analysis showed that high affinity non-functional sites are rare in the genome, meaning that most of the binding that was measured by the ENCODE project was probably functional binding. And there's a good biochemical reason for why this is the case. The first has to do with this interference effect. That is, if transcription factors were binding randomly all over the genome, that would just simply muck up the operation of the genome. If most of the binding was random binding to regions of the genome, that would create an interference that would gum up the operations associated with the genome. There's also a titration effect. In other words, there's only so many copies of a transcription factor that are going to be produced. And if these transcription factors were binding randomly to different sites in the genome, then there wouldn't be any of those transcription factors available to actually do the binding that's needed for these transcription factors to properly function. And so, in other words, there are very good biochemical reasons to think that transcription factor binding is not noise, but is actually reflecting functionality. Now, this leads us to really what is the real reason that people like Dan Grahr are actually challenging the ENCODE project. And this is what John Maddock has said, a molecular biologist. There may be another factor motivating Grahr and others, which is suggested by the sources and selection of quotations used at the beginning of the article, as well as the use of the phrase evolution-free gospel. The argument of largely non-functional genome is evoked by some evolutionary theorists in the debate against the proposition of intelligent design of life on Earth, particularly with respect to the origin of humanity. In essence, the argument posits that the presence of non-protein coding or so-called junk DNA that comprises greater than 90% of the human genome is evidence for the accumulation of evolutionary debris by blind Darwinian evolution, and argues against intelligent design, as an intelligent designer would presumably not fill the genetic instruction set with meaningless information. The argument is threatened in the face, this argument is threatened in the face of growing functional indices of non-coding regions of the genome, with the latter reciprocally used in support of the notion of intelligent design, and to challenge the conception that the natural selection accounts for the existence of complex organisms. And so, in other words, the reason, again, for challenging the ENCODE project results has more to do with really the threat of intelligent design, and the fact that, again, the ENCODE project result actually threatens to undermine key ideas in evolutionary theory. In fact, I did a debate with P.Z. Myers a few years ago at North Dakota State University on Darwin Day, and the idea of the ENCODE project came up. And PZ Myers said to me that if the ENCODE project results are correct, then it means that there are key ideas in evolution that can't be true. And so therefore, the ENCODE project results simply must not be correct. And so this highlights really what's at stake in the mind of many people. And then again, I just want to close out by just simply pointing out that in addition to the ENCODE project, there are a mounting number of studies that consistently indicate that, again, junk DNA has function. We now know that pseudogenes can code for functional proteins in some instances, and they can, again, function in a gene regulatory role through a decoy mechanism that is part of this competitive endogenous RNA hypothesis. And this, by the way, explains why pseudogenes look like the corresponding version of genes that produce produce protein products, is because their role and as in this regulatory mechanism is predicated on their similarity to pseudogenes. Now, I've had people tell me that, well, this explains functionality for pseudogenes that are transcribed, but what about the pseudogenes that aren't transcribed? With the assumption that many of the pseudogenes in the genomes aren't transcribed. Well, it turns out that assessing whether or not a pseudogene is transcribed is actually a very tricky thing, because there are researchers that are now arguing that the assays used to measure transcription may be mistaking pseudogenes for the functional version of the genes because of sequence similarities. So I don't think it's established that many pseudogenes are actually not transcribed, even though that again is the claim. Other classes of junk DNA are also functional. Line DNA plays a role in gene expression, as does sign DNA. Introns allow for alternate splicing, and they also seem to be playing a role in stress response. ERVs and EVEs play a role in have an antiviral function. They're part of the innate immune system. Tumor suppression is also something that ERVs play a role in, and elements of ERVs and EVEs play a role in gene regulation. And there's also other roles that junk DNA plays, quote unquote, that have nothing to do with an informational role, but a non-informational role. There's something known as the nucleoskeletal hypothesis that we'll talk about in a minute. Well, we'll go ahead and talk about it right now. But the idea here is that the DNA content in the nucleus actually determines the nuclear volume, and the ratio of the nuclear volume to cellular volume is a tightly constrained parameter. In other words, as the cell gets bigger, the nucleus has to get bigger in proportion, because as the cell gets larger, you have to have the ability to supply basically transcripts and other materials from the nucleus in order to sustain the activity of the cell, which requires a greater surface area of the nucleus and a greater number of nuclear pores. And so you have a very tight relationship between nuclear volume and cellular volume. And this is actually determined by the DNA content. So in other words, some of that this nucleoskeletal idea, this hypothesis, could actually explain the C-value paradox in a functional role, in a functional way, as opposed to it resolves the C-value paradox by ascribing function to DNA, to non-coding DNA. Here's a couple of diagrams. On the left, it shows the relationship between nuclear volume and cellular volume, and you can see that ratio has to be very, very tightly constrained. Between about 30%, the nuclear volume has to be about between 30% and 10% of the cellular volume in order for again, organisms to survive. And then we also see on the right-hand side, again, the correlation between nuclear DNA content and cell volume, showing that that some of the so-called non-coding DNA could be playing a nucleoskeletal role. And again, this resolves the C-value paradox in a way in which you're looking at that non-coding DNA as being functional as opposed to being the random product of an evolutionary history. In addition to that, there's some people that have argued that junk DNA may actually serve a role as a mutational buffer. Others argue that it actually plays a role in maintaining the three-dimensional structure of the genome. And this leads to a recently discovered concept known as the genome code, which basically argues that a number of the non-coding sequences in the genome are actually playing a role in helping to essentially define and establish and maintain higher order three-dimensional structures of chromosomes, which we're now learning is absolutely critical for the function of those chromosomes, in part because that three-dimensional structure is necessary to bring enhancer sequences close to the gene that is going to be transcribed and translated. These functional elements are pervasive throughout the genome, and in fact, this structural code overlaps the genetic code. The scientist Bernardi, who proposed this idea, writes, Ono, mostly focusing on pseudogenes, proposed that non-coding DNA was junk. Doolittle and Sapienza and Orgel and Crick suggested the idea of selfish DNA, mainly involving transposons visualized as molecular parasites rather than having an adaptive function for their host. In contrast, the ENCODE project claimed that the majority of the genome participated in at least one biochemical RNA and or chromatin-associated event in at least one cell type. At first sight, the pervasive involvement of isochores in the formation of chromatin domains and spatial compartments seems to leave little or no room for junk or selfish DNA. Okay, and so in other words, let's see here, let me stop sharing. In other words, this idea of most of the genome being functional is not only an idea that I think is established by the ENCODE project, but there's other independent studies that are, studies independent of the ENCODE project that are reaching comparable conclusions, that are affirming the results of the ENCODE project. And to me, the criticisms that have been leveled against the ENCODE project, for the most part, don't stand. For the most part, I think lack real rigor or lack any kind of real legitimacy.
Well, that was another fantastic presentation, Dr. Rana. I loved all your points, especially your assessment of the paper defining functional DNA elements in the human genome. I also appreciate the fact that you addressed so many of the criticisms, including this idea that low-level transcription or low-level activity must mean that the activity is spurious. And you also address pseudogenes, ERVs, and so much more. So I'd like to point out, though, that from the genetic archetype starting point and model, right, Dr. Rana, that you're proposing, you can make some really valid predictions pertaining to the function of these DNA elements, such as pseudogenes and ERVs. So the design hypothesis would then predict function, while from my understanding, the evolutionary model would predict much non-functionality. And since many of the lines of evidence used to support common ancestry, Dr. Rana, are then agnostic to the debate, as you have pointed out, the genomic archetype or common design model can equally explain the data when it comes to those lines of evidence. Therefore, my question is, could DNA function be a plausible way to help differentiate the models and tell us which one is right and which one is wrong?
Yeah, I mean, that's a great question. And, from my perspective, at least at this point, I think we're looking at, what you might say would be, in effect, a standoff. In other words, I think, the first step that we need to take as people that are ID proponents that are that advocate for kind of a creation model perspective is to simply establish the fact that a common design approach has equivalency, or is an equally valid explanation as is common descent. Because so often I hear people say that the only way to explain the nested hierarchical structures of non-functional DNA is through again common descent. But if we can again show that these sequences are functional, and we can show that there's a rationale for why these sequences bear similarity to, let's say, viral genetic material, or why pseudogenes display similarity to genes, then I think we can actually come to the table with a comparable model. Now, I at this point, the question you're right asking is the right question: is how do we distinguish between these two models? And that's that's something that I need to give a little bit more thought to. But I think by virtue of the fact that you have statements by people like P.Z. Myers, or you have people concerned about the C-value paradox being undermined by the ENCODE project results, since those are actually key, ideas in the evolutionary framework. I think the fact that they're threatened by the ENCODE project result actually tilts the scales in favor of kind of an ID approach. Something else too that I think we can make by way of prediction. And by the way, I just want to point out that in 2000, one of the very first articles I wrote at Reasons to Believe made the prediction that we would eventually discover function for virtually every class of junk DNA. This was a prediction made, again, 20 years ago, and is essentially being satisfied by fairly recent results. And I wasn't unique in making that prediction. I know a number of ID and advocates and creationists making comparable predictions. So this is a prediction that I think we want to make sure we're getting credit for at this point. But as we go into the future, what I'm noting is that our understanding of the genomes of organisms in general is in its infancy. And I suspect that when we start looking at the complexity of the gene network interactions that are taking place, that we're probably going to have to develop a systems biology approach to really understand what's going on, that it's going to become, genomics is going to become the domain of engineers, not the domain of biologists. And what I predict is that, we're going to discover that not only is our engineering principles necessary to make sense of what's genomic function, but that we're actually going to discover principles in essentially the regulation of the genome that is going to inspire new approaches to systems biology. In other words, I'm predicting that the designs that we see in genome operations are going to be so sophisticated and so impressive that it's going to inspire new technology. And so that would be a prediction that I would make now, that I think is something that I don't know that you would necessarily make from an evolutionary perspective, or at least would necessarily flow from an evolutionary perspective.
Amen. Well said. That is a fantastic prediction. So many good points you made there, Dr. Rana. It's hard to choose which one to touch on. And from my understanding and from looking into the literature, the evolutionary community has at least assumed lots of junk or evolutionary leftovers, as in the genetics of living organisms should reflect universal common ancestry. So they should find genomic fossils, and therefore they look to pseudogenes. As you pointed out, the ancient remnants of past viral infections with the ERVs and ALU sequences and so on. But I've never actually seen them, prior to discovering the incredible functionality of a lot of these DNA elements, I've never seen them make the point that this is expected or this is predicted. It's kind of like when we find this data, they retrofit it in somehow. Have you found that they expected this or even made these predictions prior to finding just how functional a lot of these, let's just say, ERVs are?
I have a similar experience as yours, where I see very little in the literature where there were any predictions about function for any of these different classes of junk DNA prior to the discovery that they indeed have function. And one criticism is that, well, what you're doing is you're taking uh, the discovery of functionality for a subset of a particular class of junk DNA as evidence that the entire class of junk DNA, all these types of junk DNA elements are functional. But the fact of the matter, it's only really a small portion or a small fraction. But this assertion is actually being undermined as we do more and more work, and as we're discovering, things like the competitive endogenous RNA hypothesis, because this is now moving from, a particular sequence has just acquired function through some kind of neo-functionalization, to actually saying, no, we actually have an elegant theoretical framework that explains why all of pseudogenes are actually functional, right? And same things with understanding endogenous retroviruses and EVEs as being part of the innate immune system. Now we have an elegant explanation for why these sequences look like viral elements, and we're seeing that more and more data is indicating more and more pervasive function for the different classes of junk DNA. It's not just an isolated case here or there. And that to me argues against neo-functionalization, where I would expect if the functionality is rare, then you could say neo-functionalization makes sense, where evolution is just using the material that's laying around and co-opting it and modifying it to create systems with new function. But if the function becomes pervasive, then this neo-functionalization argument becomes harder and harder to make, I believe.
Right, exactly. Because if what we see based on the prediction is genome-wide functionality, which the trajectory seems to favor genome-wide functionality, including these DNA elements like the ERVs, the pseudogenes, ALU sequences, then to employ neo-functionalization or co-option for such pervasive functionality in the genome, that really seems kind of like a stretch, right? Because it's
right. And to me, I think this is a good place to bring up this point too, is that many times, evolutionary biologists simply declare regions of the genome to be non-functional without actually doing any kind of experimental work.
right.
It's all based on what they expect to be the case, given evolutionary theory, right? And so, theory guides, experiments decide, and to me, nobody, in many instances, when again sequences are declared to be non-functional, people have not done the experimental work to show that's the case. It's just rather, again, an assumption that's made.
Well said, Dr. Rana. Well, here's an argument, and I appreciate how thorough you were in your presentation, because you really nailed it on a lot of these criticisms raised against ENCODE. But another argument is, they'll say because approximately 5% of the human genome is conserved across all mammals, the critics will say that since conservation is strongly indicative of function, the biochemical function that is outside of the conserved regions must then be non-functional. Have you heard this argument, and what would be the best way to respond?
Yeah, I have, and in many respects, that argument is, in a sense, very similar to the argument that Dan Grahr was making about the selected effect definition of function.
Right.
That is, in other words, if you can't show that a region is under selection, then you can't really conclude that it's functional. And so that 5% number would be a number that, Grahr, I think, argues that the maximum level of functionality reasonably in the human genome would be on the order of 25% under extreme conditions. And he argues that most reasonably it's probably about 10%. And so this is, if if it's selected, then it's going to be a region that is presumably conserved. It's a comparable idea that the conserved sequences are essentially those sequences that are under selection, and they're not able to change because they're of critical functionality. But just because you have non-conserved sequences doesn't mean that they're not functional. You just simply can't conclude that. It could very well be that they may be regions of the genome that actually can tolerate high levels of mutational change and still retain their function, particularly if those regions are actually playing a non-informational role, like let's say part of the nucleoskeletal role, or if they're playing some kind of role as a mutational buffer, as an example. Or maybe there are regions of the genome that can tolerate high levels of changes and still retain function. So, for example, regulatory elements typically can undergo a greater degree of mutation without compromising function than protein coding regions of the genome. We also are making the assumption that those differences are due to evolutionary changes. Mutational changes. It could very well be that they are intentionally designed in that way because they're performing different types of functions, though maybe related functions, but they may be functioning a little bit differently in different organisms. And so is that lack of conservation reflecting selection, some kind of selection effect, or is it actually intentionally designed that way because it needs to be different in order to perform the specific function it performs in that particular organism? So I don't think you can necessarily conclude just because something is non-conserved that it's not functional.
Great response. So many good points. And that's my thinking as well. Firstly, as you pointed out, they assume that the conserved sequences are important and functional, and therefore selection's preserving them over time, right? But as you've pointed out, the non-conserved regions can certainly be there for a number of reasons. Backup code or playing a nucleoskeletal role. And also what you kind of said there at the end, and I wonder if you would agree with this, if we as intelligent design advocates would propose special creation events, then would we then predict that some non-conserved regions are maybe just specific to different creatures that maybe aren't related, and therefore their functional roles will be specific to different creatures?
Yeah, that exactly. I would agree with that. That's definitely a possibility.
Yeah, that's a great answer. I appreciate that because, Dr. Anna, I hear that argument over and over and over again. So I'm kind of glad that we touched on it there a bit. Now, I want to point out, I've read so many of your articles. I highly recommend them. And on November 3rd, you published an article titled Endogenous Retroviruses Help Fight Tumors. And I love the way you ended the article, Dr. Anna. You pointed out that so far from being the villain of this tale, ERV sequences turn out to be P53's faithful sidekick in the battle against cancer. Can you elaborate a bit more on this article and why it's important in terms of this debate we're having?
Yeah, well, P53 is considered to be, kind of the guardian of the genome, where it's playing a number of roles that help to maintain genomic integrity. And one of the roles that it plays is actually suppression of the transcription of endogenous retroviral elements in the genome. But it turns out that when the cell is actually under stress, the proteins that work in conjunction with P53 become inactive, and this actually leads to P53 causing the transcription of endogenous retroviral elements. And what ends up happening is when these ERV elements are transcribed, it gives the cell the appearance of actually being invaded by a viral invader, having a retroviral infection, which then targets that cell for destruction by the immune system. And so it turns out that there are some tumor cells that actually can evade detection of the immune system. And so this mechanism that's mediated by P53 is a way for those tumor cells that are evading the immune system become now present, or now become detectable by the immune system, so that the immune system can clear those tumor cells out of the body. And so researchers are looking to take advantage of this fact to actually develop these P53, these drugs that will essentially stimulate P53 activity, and specifically the transcription of ERVs as an anti-cancer therapy. But in other words, what we're looking at here are these endogenous retroviral elements actually playing a role in tumor suppression by creating by essentially viral mimicry. That's I think the phenomena name given to that phenomena is viral mimicry. And so this is just, really cool discovery that shows us that, we really have misunderstood, what endogenous retroviruses are doing in cells, and that they actually, again, are playing very important functional roles. And just because, again, they're not normally transcribed doesn't mean that those ERV sequences aren't actually critical,
Right.
because normally they wouldn't be transcribed. And that would be, again, something that people would point to, saying, look, this indicates that they are indeed non-functional. But their transcription will happen only under select circumstances. But under those circumstances, that they are indeed functional. And so you can't just simply conclude because a sequence isn't transcribed, or it's transcribed at very low levels under certain cellular conditions or for certain cell types and certain tissues at certain stages in development, that these sequences are necessarily non-functional. They could very well be functional only for brief periods of time in certain cell types under certain conditions.
Now, so much amazing information. So it's safe to say then, Dr. Rana, that these two main arguments that critics of intelligent design and critics of the ENCODE project, for example, how they say that, well, low-level transcription or low-level activity means the activity must be spurious or just noise. And then at the same time, they look to the non-conserved region. So they'll say, okay, low-level activity and non-conservation means it's probably not really functional. But as you're demonstrating here, no, those are not two good criteria to determine whether something is functional or non-functional.
Right. I mean, the ultimate criteria is that you have to roll up your sleeves and do the hard experimental work.
Right.
Where, you need to really probe, whether or not indeed it's, those sequences are functional or non-functional.
Great response. And I apologize if some of this you addressed in your presentation, because it was so thorough. But as you know, the critics will point out that this transcription, which they'll say is, 60+% of the genome being transcribed, they'll say about 2/3 of this transcription is exons and introns. And since introns are non-protein coding and would make up the greater portion of that, they'll point out that introns are transcribed and then cut out of the RNA before translation occurs, right? And they will admit, from my interactions with them, they will admit that parts of the introns are functional in the sense that they serve as markers at their ends. But they'll say that the long regions, Dr. Anna, in between, the cut here markers are very messy. And they'll point to, the fact that they're full of transposons and non-conserved random sequences. And they'll say, so therefore, the majority of it is still non-functional, even if we have a little bit of a function on the ends of them. Does that make sense to you? Or, what would be a good way to respond to this?
Yeah, well, and first of all, I would just say that the presence of introns, in genes actually allows for alternate splicing, allows for that transcript to be modified in a variety of different ways. to produce, a number of different, alternate transcripts. And so one gene can actually, in effect, encode for several different types of protein products. And so when you have alternate splicing, the introns that are not removed are actually playing a role in the structural makeup of that particular protein product. Number one. Number two is that there have been studies published recently that have shown that introns that are spliced out of genes actually can go on and play other regulatory roles, in the cell. So they're not necessarily just simply RNA discards, but they actually are playing other regulatory roles, particularly playing a role in the biochemistry of the stress responses during conditions like starvation and things like that. So we're again just assuming that introns are non-functional, that they only are serving one particular role in biochemistry. And the fact of the matter is they may be doing things that we've yet to discover. And so this is really there's not experimental evidence that they're non-functional or unimportant. It's essentially an assumption being made. Whereas on the other hand, we're seeing other studies actually indicating that there's a wide range of different types of functions that introns actually assume.
I love it. It's such a great time. It's 2021 and it's so much fun because we're, as you know, I mean, there's just so much information, so many more papers and data coming out showing just how, functional portions of the genome are that were once assumed to be non-functional. As you pointed out, one gene can encode for several different protein products. And I find alternative splicing fascinating, and where the evolutionist says most of the introns are just RNA discards, as you've put it, Dr. Rana, they actually indeed do function in a variety of ways. And also we're in the infancy of understanding the DNA language in the first place, right? And that's why, as you've pointed out, we can make predictions and we can be confident about those predictions because of the trajectory. Where I'm not seeing, the evolutionary community really making predictions. I guess they're just more so attacking, the ENCODE project and just hoping for junk.
Yeah, and to be clear, there are a number of life scientists who actually do see enormous amount of value to what the ENCODE project has done, right? And the ENCODE project has again put together an impressive collection, or impressive catalog of biochemical activity that's associated with different regions of the genome that then provide a starting point, I think, for people to really begin to understand, individual, elements of the genome and whether or not they're functional and what that functional role might actually be. So, so there are scientists, to be fair, that really do see value to the ENCODE project.
Well said. Well said. Yeah, I should be more clear on that, in that we do have a lot of critics that kind of fight and attack it. But at the same time, there are a lot, and from my understanding, a lot of scientists that are involved at the NIH, National Institute for Health, that do see a lot of good out of these research programs. Here's a good question, actually, Dr. Rana. What are your thoughts on the gulo pseudogene? As we have talked about pseudogenes and these so-called genetic mistakes, but specifically the gulo pseudogene. Do you find this to be convincing evidence for human evolution and ancestry with the great apes, and kind of how do we deal with this?
Yeah, and to me, when it comes to the gulo pseudogene, the way I would respond is that the competitive endogenous RNA hypothesis essentially explains a functional role for unitary pseudogenes. Is that even though these pseudogenes may no longer produce or may not necessarily produce transcripts that are translated. Oops.
No problem.
Sorry about that.
No
Even
worries.
though they may not produce transcripts that are translated, they still are producing transcripts that are part of this this microRNA network of transcripts. And so their activity in that network is influencing the translation of other transcripts that are part of that network. So they're playing a regulatory role, even if they're not necessarily playing a role in which the information is translated. So so I don't know what specifically the function of the gulo pseudogene is necessarily, but its generalized function would be part by virtue of the fact that it's playing a role in this network.
Right. Great response. Great response. Here's a question as we start kind of winding it down. Time always flies by with you, Dr. Rana. You give us so much great information, and a lot of people in the chat, and I feel the same way, are pointing out that your presentation's rewatchable. It's going to have to require a few watches to kind of soak all the information in. This question came in from Guzman 1611. I appreciate the question, Guzman. He says, question for Dr. Rana. How should we as creationists or intelligent design advocates define information from a biological point of view?
Wow, that's a really good question. And I think that there are two different concepts of information that I think would be that are practical, or sorry, that are helpful. One would be defining information in semantic terms. And so, this would be related to kind of the idea of Shannon information, where Shannon information is just simply the probability of getting a particular sequence of elements or characters in a particular order, where the lower the probability, the higher the information content. But you can have Shannon information that has no meaning whatsoever. What is important when it comes to information in a biological context is the semantic concept, where that there are particular sequence combinations that actually harbor biochemical functionality and hence biochemical meaning. And what gives meaning to syntactical information would be the genetic code. So you can't really think about semantic information until you're starting to think about really the genetic code and the types of protein products that are being essentially that are essentially being defined, if you will, by that information found in DNA. So when you start talking about semantic information, now you're starting to talk about kind of an integrated set of systems that's giving meaning to that information. Another different approach that I think is helpful would be an algorithmic definition of information, where the algorithmic definition would be what is the set of instructions required to produce that particular sequence. Sequences that have high levels of algorithmic information are sequences that require a very complex set of instructions to actually account for the sequence, where the maximum amount of algorithmic information would be essentially the set of instructions that essentially is a delineation of the actual sequence itself. But one of the things that's really provocative is that it seems as if when you look at biomolecules, is that there's a set of algorithmic information that's instantiated within the structure of biomolecules, but that set of instructions, again, is part of an integrated system of biomolecules, where that information or those set of instructions seems to tell those molecular systems how to essentially function in conjunction and collaboration to accomplish some kind of end product. And so those are two concepts, I think, of information that are kind of distinct from each other, but are very helpful. I personally think the algorithmic information concept is the most useful and the most versatile, and also the one that I think raises the most significant challenges to an evolutionary explanation for bioinformation.
Great response to a great important question, Dr. Rana. Let's end it with this next question. And I love that because I'm looking over my notes, and you know, a lot of the criticisms that have been raised against ENCODE and arguments in support of junk DNA, and we've touched on them all. You've done a great job addressing them. Again, another one of your articles, though, that I really enjoyed, Dr. Anna, was published on October 20th, 2021. It's titled Endogenous Viral Elements, EVEs, Help Wasps to Parasitize. And in the article, you provide a response to the problem of natural evil and to bad designs. I was wondering if you could touch on this important issue, even just briefly, Dr. Anna.
Yeah. Well, I mean, you know, this, you know, the idea of junk DNA as a challenge to the design argument is really kind of an embodiment of the problem of natural evil to some degree, which is essentially, why would God create a world that looks this way? If God is all powerful, all knowing, and all good, then we would expect a world that's very different than the world that we live in. You know, why is there so much pain and suffering in nature? Why is there parasitism? Why is nature red in tooth and claw? Or why do we see flawed designs in biological systems? You know, these kind of systems, the argument would be something we'd expect evolutionary mechanisms to create, not the God of the Bible, who again is all powerful and all knowing and all good. But really what's missing in that assessment is that maybe there's actually a rationale for why the world is indeed the way that it is. And you know, there are very good reasons why, you know, parasitism exists in nature. It's a way to control numbers. And so, you know, for many of the organisms that are the prey of wasps, you know, that lay their eggs in, you know, in the bodies of caterpillars and things like that, they're actually playing a role in curtailing the numbers of these pests that would destroy, you know, plants that would destroy agricultural crops. And so you could actually see a reason why God would create a world with parasitism. And you know, what's interesting is that these parasitic wasps actually employ endogenous retroviruses as a means by which to stave off the the response of the caterpillar to the invasion of the wasps laying eggs within its body. The caterpillars will mount these immune responses against the caterpillar eggs. And so what these endogenous viral elements in the genomes of wasps do is actually become activated during the invasion and essentially produce proteins that essentially inhibit the response of the caterpillar. And so it's here that the endogenous viral elements are functioning like a viral vector that are carrying genes into the caterpillar that again are shutting down its innate immune system. But what's intriguing to me is that what these wasps are doing with endogenous viral elements is very similar to what is being explored in gene therapy today, where people are looking at the use of, you know, basically viral vectors as a way to deliver genes to cells for gene therapy. In fact, if you look at the Johnson & Johnson vaccine, the AstraZeneca vaccine, they're basically using an adenoviral vector to carry the gene that encodes for the spike protein into cells in our bodies. And so that's essentially the same kind of mechanism, interestingly enough, that these wasps are using with these endogenous viral elements. So to me, very intriguing.
Very interesting. I appreciate that detailed response, Dr. Rana. Here's a good way to end it, going right back to the main topic of tonight: junk DNA, DNA function, ENCODE, and even predictions pertaining to, you know, the evolutionary model and the genomic archetype model. Dan Straight, thank you for the question. He says, how confident are you, Dr. Rana, in the functionality of the quote unquote junk? And what percent would you consider functional overall? God bless you. Great presentation.
Yeah, that's a great question. Number one, I'm very confident in the fact that junk DNA is functional because, again, the trend line continues to move explosively towards functionality. It's in if I'm going to be honest, in 2000 when I made the prediction that junk DNA would be functional, I made that prediction based off of essentially what are the creation model that we were developing at Reasons to Believe would demand. But was I confident that that prediction would be satisfied? I don't know how confident I really was. But here we are 2 decades later, and we now are seeing functionality for things like pseudogenes. We're seeing functionality for endogenous retroviruses in ways that I don't know that I could necessarily have imagined a couple of decades ago. And this recognition that endogenous retroviral elements are part of the innate immune system is something that really in the last 5-6 years has been recognized by biologists, and now there's an explosion of papers essentially going down this path, providing example after example after example of ways that ERVs contribute to the innate immune system. And so I'm completely confident that we're going to continue to see more and more discoveries of function for junk DNA. I think we're going to see more and more studies that continue to affirm the results of the ENCODE project. We're going to see more and more instances where the results of the ENCODE project are proving to be extremely valuable for biomedicine. But what is the exact percentage of the human genome or the genomes of organisms that are functional? I don't know. I think I would suspect, even as a creationist, that there's going to be some level of non-functionality in genomes. I think that's just serves to reason, regardless of your view of how genomes have come to be. I think there's going to be some level of non-functional elements, but I don't know the precise number. I'm really letting the data make that determination. But I think at this point, what these discoveries have done is really changed the way that we're thinking about the genome. It's far from being this wasteland of junk. It looks like an incredibly elegant, incredibly sophisticated system that, again, is going to be a challenge for systems engineers to truly to decipher in terms of its functionality.
Great answer. I completely agree. Dr. Rana, so many good points, so much good information. Again, I appreciate your time. I know how busy you are. And, I thank you so much for giving us your time for what I would call this two-part series. We did part one a couple months ago. We did this one. Anybody who has not yet seen part one, please check it out. I've got it on our website, actually, right on the homepage and in the description box of this video. Last time we even dealt with things like hierarchies in genetics. We dealt with third position codon variation, ERVs, pseudogenes. So we've really touched on a lot. So I want to encourage people to please share around these videos. The truth is important. Dr. Rana, again, thank you so much. I appreciate it. Any final words, final thoughts, anything you wanted to add before we kind of wrap it up here?
Well, first of all, Donnie, thanks for having me. You've been a gracious host, and I'm, honored to be, part of what you're doing at Standing for the Truth. And so thanks for having me. And, I think the if there's a final thought, it would just simply be this, and it's a point we've already made, but just keep in mind that we really are very much in our infancy, excuse me, when it comes to studying genomes. And I think we want to be just a little bit circumspect before we start drawing conclusions, until we actually get a better understanding, a better grasp of these systems. But I also think that what we're seeing is that a creation model approach, an intelligent design approach to genomes actually is a very fruitful way of thinking about them. It leads to advances, it leads to discoveries, and I would dare say that an evolutionary paradigm has actually stymied and stultified advances in genomic science, has not actually propelled forward an understanding. Because if you deem a particular class of sequence elements to be non-functional, there's no motivation for studying them. And yet what we have discovered is simply coincidentally, by accident, is that these regions that have been deemed non-functional turn out to be actually critical, in the etiology of many diseases. And so we've really delayed important biomedical understanding because of the commitment that people have to the evolutionary paradigm, which I just don't think is a fruitful way of studying or thinking about genomes. I think, again, a creation model approach, an ID approach is a much more productive format.
Amen. Again, I agree with every single word. We, from the creation or design starting point, we expect treasure. And that's exactly what we're finding. And as you said, we're just in the infancy of understanding the DNA language. So we expect more and more treasure in the future. So again, thank you so much, Dr. Rana. God bless you. Thank you, everybody in the chat. Thank you for all your great comments, feedback, and questions. Standing for Truth is out.
I hope you enjoyed this video. Please make sure to hit that like button. It really does help. And subscribe if you enjoyed the content and want to see more.